ಗುಡ್ ಈವ್ನಿಂಗ್ ಮ್ಯಾಮ್.
ಗುಡ್ ಈವ್ನಿಂಗ್ ಅನುಷಾ.
ಗುಡ್ ಈವ್ನಿಂಗ್.
ಸರ್ ಒಂದ್ಸರಿ ಚೆಕ್ ಮಾಡಿದ್ರ?
ಹಾ ಸರ್ ಇದ್ದಾರೆ.
ಅಲ್ಲ ಅದೇ ಸ್ಕ್ರೀನ್ ಶೇರಿಂಗ್ ಎಲ್ಲ.
ಹಾ ಮ್ಯಾಮ್ ಎಲ್ಲ ಚೆಕ್ ಮಾಡಿ ಆಯ್ತು.
Okay, I have an option like present now option is there. Through that I can share.
ಗುಡ್ ಈವ್ನಿಂಗ್ ಮೇಡಮ್.
ಗುಡ್ ಈವ್ನಿಂಗ್ ಸರ್.
Yeah.
Good evening, sir. Manjula ma'am, can you hear me?
Yes, Jyoti madam.
Okay, ma'am. Okay, thank you. Good evening, Prema ma'am.
Good evening, Jyoti madam.
ಗುಡ್ ಈವ್ನಿಂಗ್ ಮ್ಯಾಮ್. ಗುಡ್ ಈವ್ನಿಂಗ್ ಸರ್.
ಕಲ್ಪನಾ ಮೇಡಮ್ ನಮಸ್ತೆ. ಮ್ಯೂಟ್ ಆಗಿದೆ.
ಕಲ್ಪನಾ ಮ್ಯಾಮ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ.
ಹಾ ಮಾಡಿದ್ದೀನಪ್ಪ. ಎಷ್ಟು ಪಾರ್ಟಿಸಿಪೇಟ್ ಕೆಳಗಡೆ ಕಾಣಿಸುತ್ತಲ್ಲ.
ಕಲ್ಪನಾ ಮೇಡಮ್.
ಹಾ ಹೇಳಿ ಪ್ರೇಮಾ.
ಸ್ವಲ್ಪ ಸ್ಟ್ರೈಟ್ ಬನ್ನಿ. ಆಫ್ ಮಾತ್ರ ಸ್ಕ್ರೀನ್ ಬರ್ತಾ ಇದೆ, ಅರ್ಧ ಮಾತ್ರ ಕಾಣಿಸ್ತಾ ಇದ್ದೀರಿ. ಅರ್ಧ ಭಾಗ ಮಾತ್ರ ಕಾಣಿಸ್ತಾ ಇದೆ, ಹಾಗಾಗಿ ಸ್ಟ್ರೈಟ್ ಆಗಿ ಕೂತ್ಕೊಳ್ಳಿ ಅಂದೆ. ಅಥವಾ ಫೋನ್ ಇನ್ ಸ್ವಲ್ಪ ಹಿಂದಕ್ಕೆ ಇಟ್ಕೊಳ್ತೀರಾ ನೋಡಿ. ಅನುಷಾ.
ಹಾ ಮ್ಯಾಮ್.
ಕಲ್ಪನಾ ಮೇಡಮ್ ಓಕೆನಾ?
ಹಾ ಮ್ಯಾಮ್. ವಿಡಿಯೋ ಏನೋ ಸ್ವಲ್ಪ ಸ್ಟಕ್
ಅದೇ
ಆಗಿದೆ.
ಸ್ವಲ್ಪ ಸೈಡ್ಗೆ ಆಗಿದೆ ಅನ್ಸುತ್ತೆ.
Prima madam.
Madam.
7 exactly commence madam. Start.
ಹಾ ಸರ್ ಇದ್ದಾರೆ, ಮೇಡಮು ಬಂದಾಗಿದೆ. ಕರೆಕ್ಟ್ 7ಗೆ ಸ್ಟಾರ್ಟ್ ಮಾಡ್ಬಿಡೋಣ ಅಥವಾ ಇನ್ 5 ಮಿನಿಟ್ಸ್ ಇದೆಯಾ? ಆಯ್ತಾ?
In 5 minutes ಇದೆ madam.
ಇನ್ 5 ಮಿನಿಟ್ಸ್ ಇದೆ. ಇನ್ನೊಂದು 2 ಮಿನಿಟ್ಸ್ ಆದ್ಮೇಲೆ ರಶ್ಮಿ ರಕ್ಷಾಗೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿಸ್ಬಿಡೋಣ. ಅವಾಗ 1-2 ನಿಮಿಷ ಆ ಕಡೆ ಈ ಕಡೆ ಆದ್ರೂ ಮ್ಯಾನೇಜ್ ಆಗುತ್ತೆ. ಅಂದ್ರೆ ಇನ್ 2 ಮಿನಿಟ್ಸ್ ಬಿಟ್ಟು.
Okay. ಆಯ್ತು.
ಪ್ರೇಮ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ಇಂದ ಯಾರಾದ್ರೂ ಜಾಯಿನ್ ಆಗಿದಾರಾ?
ಸರ್ ನಂದಿ ಸರ್ ಬರ್ತಾರಂತೆ ಮೇಡಮ್, ಸೆಕ್ರೆಟರಿ ಸರ್ ಜಾಯಿನ್ ಆಗ್ತೀನಿ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ರಕ್ಷಾ, ರಶ್ಮಿ, ರಕ್ಷಾ, ರಶ್ಮಿ ರೆಡಿನಾ?
ಮ್ಯಾಮ್ ರೆಡಿ ಇದೀವಿ.
ಮಂಜುಳಾ ಮೇಡಮ್
ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತೀನಿ.
ಕಮೆಂಟ್ಸ್ ಮಾಡ್ಲಾ ಮೇಡಮ್? ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತೀನಿ.
ಮ್ಯಾಮ್ ಒಂದ್ ನಿಮಿಷ. ಮ್ಯಾಮ್ ಒಂದ್ ನಿಮಿಷ.
ಅನುಷಾ ರೆಕಾರ್ಡಿಂಗ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿ.
ಅದೇ ಮ್ಯಾಮ್ ಒಂದ್ ನಿಮಿಷ. ಕರೆಕ್ಟಾಗಿ 7ಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಸ್ಟಾರ್ಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ಮ್ಯಾಮ್. ಸ್ಟಾರ್ಟ್ ಆಗ್ತಿದೆ.
ರೆಕಾರ್ಡಿಂಗ್ ಸ್ಟಾರ್ಟ್ ಆಗ್ತಿದ್ಯಾ?
ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತೀನಿ ಮೇಡಮ್.
ಮಾಡಿ ಮೇಡಮ್.
Hello. Good evening, everybody. Everyone, welcome to the Multidisciplinary 5-day International Faculty Development Program on Perspectives of Human Health and Environment, organized by the Science Forum of SJR College for Women. I am Manjula Essen, Assistant Professor, Department of Physics, and the Chairperson of SJR Science Forum SJR College for Women, welcoming you all to this FDP. The COVID pandemic situation has given an opportunity to think and rethink about using technology effectively, not only for online teaching, but also in conducting workshops, symposiums, strategy development programs, all together in a different way. The objective of this program is to empower the participants in understanding the importance of health and environment in the current scenario, and also the responsive steps to be followed for conservation and improvement. I now request Raksha and Rashmi of second year BCom students to invoke the blessings of God, the Supreme Being, by an invocation song. Over to Raksha and Rashmi.
महागणपत्यो मनसा महागणपत्यो मनसा स्मरामि महागणपत्यो मनसा स्मरामि वसिष्ठवामदेवादिवन्दित महागणपत्यो महादेव सुतं महादेव सुतं महादेव सुतं गुरु गोहनुतो महादेव सुतं गुरु गोहनुतो मारकोटि प्रकाशं शतं मारकोटि प्रकाशं शतं महाकविनाटकादिप्रियम् महाकविनाटकादिप्रियं मूषिकवाहनमोदकप्रियं महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दित महागणपतिं मनसा स्मरामि.
Thank you, Raksha and Rashmi, for a melodious, harmonizing song. Today, we are indeed honored and grateful to have with us Dr. Basavaraju G. Sanganahalli, neuroscientist, radiology and biomedical imaging, Yale University School of Medicine, New Haven, Connecticut, United States of America, to speak on advances in functional brain imaging. On behalf of SJR Education Society, Management, and SJR College for Women, I extend a warm welcome to Dr. Basavaraju for today's session. Welcome, sir. I cordially welcome Srimati Kalpana T.S., Principal of our sister institution, that is SJR PU College for Women, who is presiding over today's session. Welcome, Kalpana Madam. I cordially welcome our college principal, Dr. Prema Siddharaju, to this session. Welcome, Prema Madam. I extend a warm welcome to Sri N.R. Nandish sir, Secretary of SJR Education Society, to this program. Welcome, sir. Welcome, Nandish sir. I cordially welcome Sri Shiv Prakash, President, and Sri C. Natraj, Treasurer of SJR ES, to this session. I am very happy to welcome all the members of the Science Forum and the technical team for today's session. I extend a pleasant welcome to all the faculties of UG and PG departments from various colleges and regions attending this program. Now I request Dr. Jyoti, the Department of Biotechnology, to introduce the resource person of today's session. Over to you, Jyoti Madam.
Thank you, Manjula ma'am. I would like to take this opportunity to introduce our speaker for 5-day International FDP program. Dr. Basavaraju G. Sanganahalli received his PhD in neuroscience. It's audible, ma'am?
Yes, Jyoti Madam.
Okay. In neuroscience from the National Institute of Mental Health and Neurosciences in the year 2004. After his postdoctoral training at the Magnetic Resonance Research Center, Yale University School of Medicine in the areas of functional magnetic resonance imaging and neurophysics physiology to join the research faculty of radiology and biomedical imaging in the year 2008. He is one of the core scientists in the functional neuroimaging course, quantitative neuroscience with magnetic resonance. His research interests are in the development and applications of multimodal biomedical imaging methodologies to study brain function in animal models. Dr. Sanganahalli is applying this multimodal imaging tool to understand brain function in healthy as well as psychiatric or neurological disorders, as like Alzheimer's diseases, so on, in animal models. Sir has published more than 44 articles in the peer-reviewed journals with Google citation 1200 and H index 20. Sir has authored few books and 4 book chapters, and Doctor has presented more than 100 paper and posters of competitive or peer-reviewed in various international societies or conferences. Doctor has delivered more than 10 guest lectures or invited talks in various universities, to name a few: University of Delaware, USA, Washington State University, so on and so forth. Sir has received various awards and honors, and the list goes like this. To name a few: Elsevier Reviewer Recognition, Distinguished Reviewer for Magnetic Resonance and Imaging, Elsevier Reviewer Recognition, and 3 consecutive Student Stipend Award, International Society for Magnetic Resonance in Medicine, Senior Research Fellowship by Department of Science and Technology, Government of India. Sir's teaching and mentoring experiences includes teaching for MPhil students in biophysics in National Institute of Mental Health and Neurosciences, teaching for MS Biotechnology and undergraduate biology students biophysical tools to study neuronal and astrocytic cells, teaching for UG students in physics at SJR College, Bangalore, mentoring 3 PhD students and many UG and master students. Sir's professional and academic services again is too long. To name a few, he has been a present member for peer reviewing, also member for various organizations, and Sir has served as reviewer for grant agencies like Health Research Board, King's College London, Alzheimer's Drug Discovery Foundation, Alzheimer's Association, American Heart Association. Doctor is member of various professional societies. Again, the list is long. To name a few: Alzheimer's Association, American Heart Association, International Society for Magnetic Resonance in Medicine. I would be glad to welcome such a wonderful guest speaker for this FDP program. Welcome, sir.
So should I present now?
Thank you, Dr. Jyoti. Sir, we have a keynote address by our principal, sir.
Okay.
So now I request Dr. Prema Siddharaju, Principal, SJR College for Women, for the keynote address. Over to you, Prema.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಈ ದಿವಸ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ಮಹಿಳಾ ಕಾಲೇಜು 5 ದಿನಗಳ ಇಂಟರ್ನ್ಯಾಷನಲ್ ಫ್ಯಾಕಲ್ಟಿ ಡೆವಲಪ್ಮೆಂಟ್ ಪ್ರೋಗ್ರಾಮ್ ಅನ್ನ ಪ್ರಾರಂಭ ಮಾಡ್ತಾ ಇದೆ. ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಈ ದಿವಸ ರಿಸೋರ್ಸ್ ಪರ್ಸನ್ ಆಗಿ ಭಾಗವಹಿಸ್ತಾ ಇರುವಂತಹ ಡಾಕ್ಟರ್ ಬಸವರಾಜು ಗಂಗಣ್ಣ ಸಂಗನಹಳ್ಳಿ ಅವರೇ, ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಅಧ್ಯಕ್ಷತೆಯನ್ನ ವಹಿಸಿರುವಂತಹ ನಮ್ಮ ಸಹೋದರ ಸಂಸ್ಥೆಯ ಪ್ರಾಂಶುಪಾಲರಾದಂತಹ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ಪದವಿ ಪೂರ್ವ ಮಹಿಳಾ ಕಾಲೇಜಿನ ಪ್ರಾಂಶುಪಾಲರಾದಂತಹ ಪ್ರೊಫೆಸರ್ TS ಕಲ್ಪನಾ ಮೇಡಮ್ ಅವರೇ, ಪ್ರಪಂಚದ ಮೂಲೆ ಮೂಲೆಗಳಿಂದ ಈ ಒಂದು FTP ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸ್ತಾ ಇರುವಂತಹ ಹಲವಾರು ಅಧ್ಯಾಪಕ ಮಿತ್ರರೇ, ಸಂಶೋಧನಾ ವಿದ್ಯಾರ್ಥಿನಿಯರೇ ಮತ್ತು ನನ್ನ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರೇ, ಇದು ನಮ್ಮ ಒಂದು ಅತ್ಯುತ್ತಮ ಪ್ರಯತ್ನ ಅಂತ ನಾನು ಅಂದ್ಕೊಳ್ತೀನಿ. ಯಾಕೆ ಅಂದ್ರೆ ಈ ದಿವಸ ಇತ್ತೀಚೆಗೆ BSc ಓದುವವರ ಸಂಖ್ಯೆ ಕಡಿಮೆ ಆಗ್ತಾ ಇದೆ. ಎಲ್ಲ ಇಂಜಿನಿಯರಿಂಗು, ಮೆಡಿಕಲ್ಲು ಅಂತ ಹೇಳಿ ಸೈನ್ಸ್ ಓದುವಂತವ್ರು ಹೋಗುವಾಗ BSc ಮಾಡಿಕೊಂಡು ಅದರಲ್ಲೂ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆಯಲ್ಲಿ BSc ಓದಿ MSc ಡಾಕ್ಟರೇಟ್ ಮಾಡಿಕೊಂಡು ಪ್ರಪಂಚಕ್ಕೆ ಒಂದು ಒಳ್ಳೆ ಕೊಡುಗೆಯಾಗಿರುವಂತಹ ಬಸವರಾಜು ಸಂಗನಹಳ್ಳಿ ಅಂತವರು, ನಾಳೆ ಮಾತಾಡಲಿರುವಂತಹ ನ್ಯೂಜಿಲೆಂಡ್ ನ ಮಂಜುನಾಥ್ ರೆಡ್ಡಿ ಅವರು ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ಕಾಲೇಜಿನಲ್ಲಿ BSc ಪದವಿಯನ್ನ ಪಡ್ಕೊಂಡಂಥವ್ರು. ಇವರೆಲ್ಲ ಇವತ್ತು ಪ್ರಪಂಚಕ್ಕೆ ಒಂದು ಒಳ್ಳೆಯ ಕೊಡುಗೆಯಾಗಿ ನಿಂತಿದ್ದಾರೆ. ಅದು ಸಾಧ್ಯ ಆಗಿರೋದು BSc ಪದವಿಯ ಕಾರಣಕ್ಕೆ. ಸೊ ಇದು ಉದ್ದೇಶ. BSc ಯಾವತ್ತಿಗೂ ಕೂಡ ಸಂಶೋಧನಾ ಕ್ಷೇತ್ರಕ್ಕೆ ಅತ್ಯಂತ ಅಡಿಪಾಯವಾಗಿ ಬೇಕಾಗಿರುವಂತದ್ದು ಅನ್ನೋದನ್ನ ಒಂದು ಮನದಟ್ಟು ಮಾಡಿಕೊಡೋದಕ್ಕೆ ಇವರಿಬ್ರು ನಮಗೆ ಮಾದರಿಯಾಗಿ ನಿಂತಿರೋ ಅಂತವ್ರು. ಇವರು ಮಾತ್ರ ಅಲ್ಲ, ನಂತರದ 3 ದಿವಸಗಳಲ್ಲಿ ಒಟ್ಟು 5 ದಿವಸಗಳು ಏನು ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ ನಮ್ಮ ಈ FDP ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ, ಇವರೆಲ್ಲ BSc ಪದವಿಯನ್ನ ಪಡ್ಕೊಂಡು ಹೇಗೆ ಅವರದೇ ಆದಂತಹ ಸಂಶೋಧನಾ ಕ್ಷೇತ್ರದಲ್ಲಿ ತಮ್ಮನ್ನ ತೊಡಗಿಸ್ಕೊಂಡಿದ್ದಾರೆ, ಇದು ಇತರರಿಗೆ ಮಾದರಿಯಾಗ್ಲಿ ಅನ್ನುವಂತಹ ಒಂದು ದೊಡ್ಡ ಉದ್ದೇಶ ಕೂಡ ನಮ್ಮದು ಆಗಿದೆ. ಈ ಒಂದು ವೆಬಿನಾರ್ ನಲ್ಲಿ ನಮ್ಮ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆಯ ಸುಮಾರು 6 ಕಾಲೇಜುಗಳು, 1 ಲಾ ಕಾಲೇಜು, 2 ಪದವಿ ಕಾಲೇಜು ಮತ್ತು 3 PUC ಕಾಲೇಜಿನ ಪ್ರಾಂಶುಪಾಲರುಗಳನ್ನ ಅಧ್ಯಕ್ಷತೆಗೆ ಕೇಳ್ಕೊಂಡಾಗ ಅವರೆಲ್ಲರೂ ಕೂಡ ಸಂತೋಷದಿಂದ ಈ 5 ದಿನಗಳ ಒಂದು FDP ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸಂತೋಷವಾಗಿ ಒಪ್ಕೊಂಡು ಪಾಲ್ಗೊಳ್ತಾ ಇದ್ದಾರೆ. ಜೊತೆಗೆ ಇಂಥ ಒಂದು ಮಹತ್ತರವಾದಂತಹ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನ ನಡೆಸೋದಕ್ಕೆ ನಮ್ಮ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆಯ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಶಿವಪ್ರಕಾಶ್ ಅವರು, ಸೆಕ್ರೆಟರಿಗಳಾದಂತಹ ನಂದೀಶ್ ಅವರು ಮತ್ತೆ ಖಜಾಂಚಿಗಳಾದಂತಹ ನಟರಾಜ್ ಸರ್ ಅವರು ಸಂಪೂರ್ಣವಾದ ಬೆಂಬಲವನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ಆ ಒಂದು ಬೆಂಬಲದ ಹಿನ್ನೆಲೆಯಲ್ಲಿ ಇವತ್ತು ಇಂತಹ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನ ಇವತ್ತು ಪ್ರಾರಂಭ ಮಾಡಿದ್ದೇವೆ. ಇದು ಇನ್ನ ಉಳಿದ 4 ದಿವಸಗಳು ಕೂಡ ನಡ್ಕೊಂಡು ಹೋಗುತ್ತೆ. ಇವತ್ತು ಒಂದು ವಿಶೇಷವಾದಂತಹ ಟಾಪಿಕ್ ಅನ್ನ ಇಟ್ಕೊಂಡು ಬಸವರಾಜು ಸರ್ ಮಾತಾಡ್ಲಿಕ್ಕೆ ಹೊರಟಿದ್ದಾರೆ. ನಿಮಗೆಲ್ಲ ಗೊತ್ತಿರುವಂತದ್ದು ಪಂಚೇಂದ್ರಿಯಗಳ ಗ್ರಹಣ. ನಾವು ಹೇಗೆ ಈ ನಮ್ಮ ನಾಲಿಗೆ, ಕಣ್ಣು, ಮೂಗು, ಬಾಯಿ ಹೇಗೆಲ್ಲ ಅವುಗಳದೇ ಆದಂತಹ ರುಚಿಗಳನ್ನ ಟೇಸ್ಟ್ ಮಾಡ್ತವೆ ಅನ್ನುವಂಥದ್ದು, ಪತ್ತೆ ಹಚ್ಚುತ್ತವೆ ಅನ್ನುವಂಥದ್ದು ಮೇಲ್ನೋಟಕ್ಕೆ ನಮಗೆ ಗೊತ್ತು. ಬಟ್ ಮೂಲಭೂತವಾಗಿ ಈ ರುಚಿ ನಾಲಿಗೆದಲ್ಲ, ಈ ಒಂದು ನೋಟ ಕಣ್ಣಿನದಲ್ಲ, ಈ ಒಂದು ಧ್ವನಿ ಕಿವಿಯದ್ದಲ್ಲ, ಇದನ್ನೆಲ್ಲ ಮೂಲವಾಗಿ ಗ್ರಹಿಸುವಂತದ್ದು ಮೆದುಳು ಅನ್ನುವಂಥದ್ದನ್ನ ವಿಜ್ಞಾನ ಕ್ಷೇತ್ರ ಈಗಾಗ್ಲೇ ಪತ್ತೆ ಹಚ್ಚುತ್ತಾ ಇರುವಂತದ್ದು. ಈ ಪತ್ತೆ ಹಚ್ಚುವ ಕೆಲಸ ಮೆದುಳಿಗೆ ಸಾಧ್ಯ ಆಗತ್ತೆ ಅನ್ನೋದಕ್ಕೆ ಸಂಬಂಧಪಟ್ಟ ಹಾಗೆ ಹಲವಾರು ಲೇಖನಗಳು ಇವಾಗ ಬಂದಿವೆ, ಸಾಕಷ್ಟು ಬೇರೆ ಬೇರೆಯವರು ಬರೆದಿದ್ದಾರೆ. ಆದ್ರೆ ಹೇಗೆ ಮೆದುಳಿಗ ಯಾವ ಭಾಗ ಯಾವ ಗ್ರಹಣವನ್ನ ಗ್ರಹಿಸುತ್ತೆ, ಆಮೇಲೆ ಯಾವುದಕ್ಕೆ ಯಾವ ಪ್ರಮಾಣದಲ್ಲಿ ಗ್ರಹಿಸೋದಕ್ಕೆ ಸಾಧ್ಯ ಆಗತ್ತೆ ಅನ್ನುವಂತಹ ಒಂದು ಅಧಿಕೃತವಾದಂತಹ ಮಾಹಿತಿ ಇದುವರೆಗೆ ನಮ್ಗೆ ಯಾರಿಗೂ ಕೊಡೋಕ್ ಸಾಧ್ಯ ಆಗಿಲ್ಲ. MRI ಸ್ಕ್ಯಾನಿಂಗ್ ಮುಖಾಂತರ ಇದನ್ನ ಪತ್ತೆ ಹಚ್ಚೋದ್ರ ಮುಖಾಂತರ, ಜೊತೆಗೆ ಇದರ ಯಾವ ಯಾವ ಭಾಗಗಳು ಹೇಗೆಲ್ಲ ಕಾರ್ಯವನ್ನ ಮಾಡ್ತವೆ ಅನ್ನುವಂತಹ ಕ್ಷೇತ್ರದಲ್ಲಿ ವಿಶೇಷವಾಗಿ ತೊಡಗಿಸ್ಕೊಂಡು ಸಂಶೋಧನೆಯನ್ನ ಮಾಡ್ತಾ ಇಡೀ ಪ್ರಪಂಚಕ್ಕೆ ತಮ್ಮದೇ ಆದಂತಹ ಕೊಡುಗೆಯನ್ನ ಕೊಡ್ತಾ ಇರುವಂತಹ ಕರ್ನಾಟಕದ ಹೆಮ್ಮೆ ಡಾಕ್ಟರ್ ಬಸವರಾಜು ಗಂಗಣ್ಣ ಸಂಗನಹಳ್ಳಿಯವರು. ಇವರು ನಮ್ಮ ಸಂಸ್ಥೆಯ ವಿದ್ಯಾರ್ಥಿ ಅನ್ನುವ ಹೆಮ್ಮೆ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆಗೆ. ಇಂಥ ಹಲವಾರು ವಿಜ್ಞಾನಿಗಳನ್ನ ಪ್ರಪಂಚಕ್ಕೆ ಕೊಟ್ಟಿರುವಂತಹ ಕೊಡುಗೆ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆಯದ್ದು. ಆ ಹೆಮ್ಮೆ ಇವತ್ತು ಪ್ರಪಂಚಕ್ಕೆ ಒಂದು ಹೊಸ ಚಿಂತನೆಯನ್ನ, ಹೊಸ ಸತ್ಯಾಂಶವನ್ನ ಹೇಳಲಿಕ್ಕೆ ತಮ್ಮನ್ನ ತಾವು ತೊಡಗಿಸ್ಕೊಂಡಿದ್ದಾರೆ. ಇವರು ಬರೀ ವಿಜ್ಞಾನಿ ಮಾತ್ರ ಅಲ್ಲ, ಇವ್ರ ಜೊತೆ ಮಾತಾಡ್ತಾ ಹೋದ್ರೆ ಪುರಾಣಗಳನ್ನ ಹೊಸ ದೃಷ್ಟಿಯಿಂದ ನೋಡುವಂತಹ ವಿಚಾರಗಳು, ವೇದ ಆಗಮ ಉಪನಿಷತ್ತುಗಳನ್ನ ಮಾತಾಡ್ತಾ ಗಂಟೆಗಟ್ಲೆ ಅವ್ರದೇ ಆದಂತಹ ಚಿಂತನೆಗಳು, ಅಲ್ಲಿ ಹೇಗೆ ವಿಜ್ಞಾನ ಮೈಗೂಡಿದೆ ಅನ್ನುವಂಥದ್ದನ್ನ ಹೇಳ್ತಾ ಹೋಗ್ತಾರೆ. ಅಷ್ಟು ಮಾತ್ರ ಅಲ್ಲ, ಇಲ್ಲಿರುವಂತಹ ಕೆಲವೊಂದು ಯೋಗದಿಂದ ಹಿಡಿದು ಪುರಾಣದಲ್ಲಿರುವಂತಹ ಕೆಲವೊಂದು ವಿಚಾರಗಳನ್ನು ಒಳಗೊಂಡು, ಅದರಲ್ಲೂ ವಿಶೇಷವಾಗಿ 12ನೇ ಶತಮಾನದ ವಚನಗಳಲ್ಲಿ ವಿಜ್ಞಾನ ಹೇಗೆ ಸಮ್ಮಿಲಿತವಾಗಿದೆ ಅನ್ನುವಂಥದ್ದನ್ನ ಗಂಟೆಗಟ್ಲೆ ಚರ್ಚೆ ಮಾಡುವಂಥವ್ರು. ಅಂದ್ರೆ ಸಾಕಷ್ಟು ವರ್ಷಗಳ ಕಾಲ ಇಲ್ಲಿಂದ ಬಿಟ್ಟು ಅಮೆರಿಕಾದಲ್ಲಿ ಅವರು ನೆಲೆಸಿದ್ದಾಗ್ಯೂ ಕೂಡ ಇಲ್ಲಿಯ ನೆಲಮೂಲ ಸತ್ವಗಳನ್ನ, ಇಲ್ಲಿಯ ಸಂಸ್ಕೃತಿಯನ್ನ, ಇಲ್ಲಿಯ ಚಿಂತನೆಯನ್ನ ಅವರು ಎಷ್ಟು ಅಚ್ಚುಕಟ್ಟಾಗಿ ಮೈಗೂಡಿಸ್ಕೊಂಡು ಇಲ್ಲಿರುವ ತಾತ್ವಿಕತೆಯನ್ನ, ಇಲ್ಲಿರುವಂತಹ ವಿಜ್ಞಾನವನ್ನ ನಮಗೆ ಅಲ್ಲಿಂದ ಪರಿಚಯ ಮಾಡಿಸುವಂತಹ ಒಂದು ಕೆಲಸವನ್ನು ಕೂಡ ಅವ್ರ ಜೊತೆ ನಾನು ಸಾಕಷ್ಟು ಸಾರಿ ಮಾತಾಡುವಾಗ ಗಂಟೆಗಟ್ಲೆ ನಿರರ್ಗಳವಾಗಿ ಇಲ್ಲಿಯ ಕೆಲವೊಂದು ವೇದ ಉಪನಿಷತ್ತುಗಳು, ಪುರಾಣಗಳಲ್ಲಿರುವಂತಹ ತತ್ವ ಚಿಂತನೆಗಳನ್ನ, ವಿಜ್ಞಾನವನ್ನ ನಮಗ್ ಮನವರಿಕೆ ಮಾಡ್ಕೊಡ್ತಾ ಇದ್ದಾರೆ ಅಲ್ಲಿ. ಸೊ ಅಂತ ಈಗ ನಾವು ನೀವು ಗಮನಿಸಿರ್ಬಹುದು, ನಮ್ಮ ವಿದ್ಯಾರ್ಥಿನಿಯರು ಹಾಡುವಾಗ ನಾವೆಲ್ಲ ಕೂತ್ಕೊಂಡು ಕೇಳ್ತಾ ಇದ್ವಿ, ಅವ್ರು ಸಡನ್ ಆಗಿ ಎದ್ದು ನಿಂತ್ಕೊಂಡ್ರು. ಇದು ನಮ್ಮ ಸಂಸ್ಕೃತಿಗೆ ಅಲ್ಲಿಂದ ಅವರು ಕೊಡ್ತಾ ಇರುವಂತಹ ಗೌರವ. ಈ ರೀತಿಯ ಸಂಸ್ಕೃತಿಯ ಒಂದು ಗೌರವಾನ್ವಿತ ವ್ಯಕ್ತಿ ನಿಜಕ್ಕೂ ಚಪ್ಪಾಳೆ ತಟ್ಲೇಬೇಕು ನಾವು. ಯಾಕಂದ್ರೆ ನಾನು ಗಮನಿಸ್ತಾ ಇದ್ದೆ, ಅವರು ಆ ಒಂದು ಭಕ್ತಿಯನ್ನ, ಆ ಒಂದು ಇಲ್ಲಿಯ ಸಂಸ್ಕೃತಿಯನ್ನ ಮೈಗೂಡಿಸ್ಕೊಂಡು, ನಾನು ಇನ್ನೊಂದು ವಿಚಾರ ಹೇಳಲೇಬೇಕು. ನಾವು ಏನೆಲ್ಲ ಪಿಜ್ಜಾ, ಬರ್ಗರು, ಮತ್ತೊಂದು ಮಗದೊಂದು ಅಂತ ನಾವು ನಮ್ಮ ಜೀವನ ಶೈಲಿಯನ್ನ ಹಾಳು ಮಾಡ್ಕೊಳ್ತಾ ಇದ್ದೀವಿ. ಅವ್ರು ನನಗ್ ಮಾತಾಡ್ತಾ ಹೇಳ್ತಾ ಇದ್ರು, ನೀವು ನಂಬ್ತಿರೋ ಬಿಡ್ತಿರೋ, ವಾರದಲ್ಲಿ 3 ದಿವಸ ಇವತ್ತಿಗೂ ಕೂಡ ನಮ್ಮ ಮನೆಯಲ್ಲಿ ಮುದ್ದೆ ಉಪ್ಸಾರು ಕಂಪಲ್ಸರಿ ಮಾಡಿ ತಿಂತೀವಿ ಅಂತ. ಇದಕ್ಕಿಂತ ಇನ್ನೇನ್ ಬೇಕು? ಇಲ್ಲಿಯ ಆ ಸಂಗನಹಳ್ಳಿಯ ಸತ್ವ ಅಮೆರಿಕಾದಲ್ಲಿ ಅಳವಡಿಕೆ ಆಗತ್ತೆ ಅಂದ್ರೆ ಇದಕ್ಕಿಂತ ಇನ್ನೇನ್ ಬೇಕು? ನಾವು ನಿಜಕ್ಕೂ ನಮ್ಮಲ್ಲಿರುವಂತಹ ತತ್ವವನ್ನ, ಸತ್ವವನ್ನ, ಸಂಸ್ಕೃತಿಯನ್ನ ಎಲ್ಲೋ ಒಂದ್ ಕಡೆ ಬಿಡ್ತಾ ಇದ್ದೀವಿ. ಆದ್ರೆ ಅನಿವಾಸಿ ಭಾರತೀಯರಾದ ಆ ಭಾರತೀಯರು ಅಲ್ಲಿ ಅವುಗಳನ್ನ ಅಳವಡಿಸ್ಕೊಂಡು ನಮಗೆ ಮಾದರಿಯಾಗಿ ನಿಲ್ತಾ ಇದ್ದಾರೆ. ಅದಕ್ಕಿಂತ ಹೆಚ್ಚಾಗಿ ಇವತ್ತು ಏನು ಅವರು ಮಾತಾಡುವಂತಹ ವಿಚಾರ ಇದೆ, ಆ ಒಂದು ಹೊಸ ಚಿಂತನೆ ಇಡೀ ಪ್ರಪಂಚದ ವಿಜ್ಞಾನಿಗಳಿಗೆ ಮಾದರಿಯಾಗಿ ನಿಲ್ಲುವಂತಹ ಚಿಂತನೆ ಆಗಿದೆ. ನಮ್ಮವರು ಹೋಗಿ ಅಲ್ಲಿ ಮಾಡ್ತಾ ಇರುವಂತಹ ಸಂಶೋಧನೆಯನ್ನ ನಮ್ಮ ಇಲ್ಲಿಯ ವಿದ್ಯಾರ್ಥಿನಿಯರಿಗೆ ಅಥವಾ ನಮ್ಮ ಪ್ರಾಧ್ಯಾಪಕರಿಗೆ, ನಮ್ಮ ಸಮುದಾಯಕ್ಕೆ ಅದನ್ನ ತಿಳಿಸ್ಕೊಡುವಂತಹ ನಮ್ಮ ಈ ಒಂದು ಪ್ರಯತ್ನಕ್ಕೆ ಅವರು ಒಪ್ಪಿದ್ದಾರೆ. ಇವತ್ತು ಈ ಕಾರ್ಯಕ್ರಮವನ್ನ ನಡೆಸ್ಕೊಡೋದಕ್ಕೆ ದೊಡ್ಡ ಮನಸ್ಸು ಮಾಡಿ, ಬಿಡುವಿಲ್ಲದ ಕೆಲಸ. ಅವ್ರು ಶ್ರೀಮತಿಯವರು ಡಾಕ್ಟ್ರು. ಈಗ ಕೊರೋನಾ ಸಂದರ್ಭದಲ್ಲಿ ಹಗಲು ರಾತ್ರಿ ಅವರು ಕೆಲಸ ಮಾಡ್ತಾ ಅವರನ್ನ ತೊಡಗಿಸ್ಕೊಂಡಿದ್ದಾರೆ. ಅಂಥದ್ರ ಮಧ್ಯೆ ಇವರು ಮಕ್ಕಳನ್ನ ನೋಡ್ಕೊಳ್ಳೋ ಜವಾಬ್ದಾರಿ, ಅವರ ಉದ್ಯೋಗದ ಜವಾಬ್ದಾರಿ, ಯೂನಿವರ್ಸಿಟಿ ಕೆಲಸದ ಮಧ್ಯೆ ನಮ್ಗೆ ಬಿಡುವು ಮಾಡ್ಕೊಂಡು ನಮಗೋಸ್ಕರ ಈ ಒಂದು ಸಮಯವನ್ನ ಕೊಟ್ಟು ಈ ಒಂದು ವೆಬಿನಾರ್ ನಡೆಸ್ಕೊಡೋದಕ್ಕೆ ಒಪ್ಕೊಂಡು ಬಂದಿದ್ದಾರೆ. ಅವ್ರಿಗೆ ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ವಿದ್ಯಾಸಂಸ್ಥೆ, ಶ್ರೀ ಜಗದ್ಗುರು ರೇಣುಕಾಚಾರ್ಯ ಮಹಿಳಾ ಕಾಲೇಜು ಮತ್ತು ನಮ್ಮ ವಿಜ್ಞಾನ ವೇದಿಕೆ ಎಲ್ಲರ ಪರವಾಗಿ ನಾನು ಅತ್ಯಂತ ತುಂಬ ಹೃದಯದ ಕೃತಜ್ಞತೆಯನ್ನ ನಾನು ಅವರಿಗೆ ಈ ದಿವಸ ಸಲ್ಲಿಸ್ತೇನೆ. ಮತ್ತೆ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮ 5 ದಿವಸಗಳು ಯಶಸ್ವಿಯಾಗಿ ನಡೀಲಿ ಅಂತ ನಮ್ಮ ವಿಜ್ಞಾನ ವೇದಿಕೆ ಸೈಂಟಿಯಾ ಏನಿದೆ ಅವರೆಲ್ಲರ ಪರಿಶ್ರಮಕ್ಕೆ ಒಂದು ಒಳ್ಳೆಯ ಬೆಲೆ ಅನ್ನೋ ಹಾಗೆ ಸುಮಾರು 500-600 ಜನ ರಿಜಿಸ್ಟ್ರೇಷನ್ ಮಾಡಿಸಿರುವಂತದ್ದು, ಸಾಕಷ್ಟು ಜನ ಇಲ್ಲಿ ಭಾಗವಹಿಸ್ತಾ ಇರುವಂತದ್ದು, ಜೊತೆಗೆ ಲೈವ್ ಸ್ಟ್ರೀಮ್ ನಲ್ಲಿ ಕೂಡ ಸಾಕಷ್ಟು ಜನ ಇರುವಂತದ್ದನ್ನ ನಾವು ಗಮನಿಸ್ತಾ ಇದ್ದೇವೆ. ಸೊ ಬಂದಿರುವಂತಹ ಎಲ್ಲರಿಗೂ ಕೂಡ ಇದು ನಿಜಕ್ಕೂ ಒಂದು ಬೆಳಕಿನ ಹೊಸ ಮಾರ್ಗವನ್ನ ತೆರೆಯುತ್ತೆ. ಈ ಒಂದು FDP ಕಾರ್ಯಕ್ರಮ ಇದರಿಂದ ಸಾಕಷ್ಟು ಪ್ರಯೋಜನ ಆಗತ್ತೆ ಅನ್ನುವಂತ ಒಂದು ಮಾತನ್ನ ಹೇಳ್ತಾ, ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನ ಒಪ್ಪಿ ಬಂದಿರುವಂತಹ ಬಸವರಾಜು ಸರ್ಗೆ, ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಅಧ್ಯಕ್ಷತೆಯನ್ನ ವಹಿಸೋದಕ್ಕೆ ಒಪ್ಕೊಂಡಂತಹ ನಮ್ಮ PUC ಕಾಲೇಜಿನ ಪ್ರಾಂಶುಪಾಲರಾದಂತಹ ಕಲ್ಪನಾ ಮೇಡಮ್ ಕೂಡ ನಾನು ಧನ್ಯವಾದವನ್ನ ಹೇಳ್ತಾ, ಹೆಚ್ಚು ಸಮಯವನ್ನ ನಾನು ತೆಗೆದುಕೊಳ್ಳದೆ ಬಸವರಾಜು ಸರ್ ಅವರ ಚಿಂತನೆಗಳನ್ನ ಕೇಳೋಣ, ವಿಜ್ಞಾನದ ತಂತ್ರಜ್ಞಾನದ ವಿಚಾರಗಳನ್ನ ಕೇಳೋಣ ಅಂತ ಹೇಳಿ ನನ್ನ ಮಾತನ್ನ ಮುಗಿಸ್ತೇನೆ. ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ.
Should I share my screen now?
Manjula ma'am, unmute.
Yes, sir. Yeah, present your screen, sir.
ಮಂಜುಳಾ ಮೇಡಮ್.
Ma'am, unmute ಮಾಡಿ ma'am.
ಮಂಜುಳಾ ಮೇಡಮ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿ.
Can you see my screen now?
Yes, sir. Yeah, it is. Yes, sir. It is.
ಅನುಷಾ,
ಹಾ ma'am.
ಮಂಜುಳಾ ಮೇಡಮ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿ. ಸ್ವಲ್ಪ ಪ್ರಾಬ್ಲಮ್ ಆಯ್ತು ಅನ್ಸುತ್ತೆ.
Yes, sir. Yeah.
ಅದೇ ಇದೆ ma'am.
Actually, if you guys are seeing the screen, can I start my presentation?
Yes, sir. Sure. It is. Seen, sir.
Okay. First of all, thank you everyone, and thank you for the kind introduction and very kind words. I'm really overwhelmed with the introduction. Being part of SJRCW, it gives me a great pleasure to give lecture on this occasion, where I had teachers in learning the fundamentals of physics, mathematics, and electronics at SJR College, and all were great teachers who inspired me to do research. Well, without taking further time, I would like to start this topic. The title of my talk today is Advances in Functional Brain Imaging. Well, we all know this is one of the vast topic, but we can speak for years. But today, I would like to tell you the three stories. In the first part of my talk, I'm going to show you how many different multimodal neuroimaging techniques are currently used to map brain function. In that specifically, I'm going to show high-field functional magnetic resonance imaging is applied in different rodent sensory model and how we can map the cortical and subcortical activations in rodent brain by this tool technique. In the second part of my talk, I will introduce this technique called calibrated functional magnetic resonance imaging. So traditionally, we use fMRI to map the brain activity. For example, if you're listening to this audio or lecture, you're looking at the slides, your visual cortex is active, and you're listening, your auditory cortex is active. We can map this activation pattern. But we all know that fMRI measures the brain activity indirectly. So this activation hues, what you see, the color blobs, can tell you the indirect activity in those localized activations. But over the years, we can extract a lot of brain energy metabolism information if we calibrate this technique by measuring another multimodal techniques like cerebral blood flow and cerebral blood volume. So we spent a lot of time on developing this technique at Yale with wonderful colleagues, and it has been applied in rodents, and we published a lot of papers on that. So now we are applying this technique in humans too, so that we can actually quantitate real activation of neurons or energy-based informations. In this, I am going to give an example how we can apply this calibrated fMRI to measure the cortical layer specific activity. That means human cortex or animal cortex has 6 different layers of cells. Each layer have a distinct population of cellular activity and different connection. We can actually elucidate by using this cool technique, and then later we can actually do some neural activity measurement and validate that. In the last part of my talk, I will show some of the animal models in neurological and psychiatric disorders. I'm currently using different models of aging, Alzheimer, Parkinson's, stroke, TBI, many different models. I will today talk about few examples for that model of Alzheimer disease, schizophrenia, and my TBI. Animal models are very valuable in fundamental neuroscience research. For example, where we create a model based on the human disease, and here we can do much more than the real in vivo human imaging experiment. That means we can do some histological measurement, electrophysiology, autoradiography, and many more. So it not only gives to identify a suitable biomarker of the disease, it helps us to understand the fundamental mechanism of the disease at cellular and molecular level, so that we can develop these drugs to see the disease prognosis, which has mostly like a translational neuroimaging and translational significance. Well, we all know that human brain is one of the most fascinating and incredibly complex organ. Even though it weighs only 2% of the body weight, it nearly consumes 20% of the total oxygen inhaled. It weighs around basically 1.5 kg, or 3 pounds of this massive jelly actually contains 100 billions of neurons. And each neuron makes around 10,000 connections with other neurons, making the network incredibly more complex. If you take this cellular network, the brain has 2 different populations of cells, the neurons and the glial cells. The glial cells were, for decades, they were considered just only back supporting actors. There are 3 different types: astroglia, microglia, and oligodendrocytes. But the more importance were given to neuron, because the neurons has a cell body and which has an axon, which make a synapse with the other neuron. The dendrites of the other neuron receives the information. If I zoom out this synapse here in the right corner, whenever there is a localized brain activity, there is an action potential. It travels along the presynaptic terminal. There's a depolarization of this presynaptic terminal, wherein calcium influx occurs, and it actually releases the stored neurotransmitters in the vesicle into the synaptic cleft. So these, for example, instance like glutamate, which is released into the cleft, which binds to the postsynaptic receptors and continue the signaling. All of this requires a lot of energy in the form of ATP. So this is basically a restless brain. It's always active, whether waking, dreaming, sleeping, running. It's always active. It has never been active. So where does the energy come from? The main energy supply for the brain is the oxidation of glucose and the blood flow, so which consumes enormous amount of energy in the form of ATP. To just give an example, you take this simple mathematical modeling. It's based on some of the experimental evidences. Here is a dendrite and neuronal cell body conducting an action potential. If you take a 1 milliliter of human cortical tissue, which consumes around 10 to the 17 ATP per second, if you can extrapolate it to the whole cortical gray area, it consumes 10 to the power 20 ATP per second. If for an adult brain, for 24 hours, it consumes nearly 10 power 25 ATP per second. Such an incredibly complex energy-based information. So currently, there are many multimodal neuroimaging techniques that have been used. They are spanning from different temporal resolution, as you see in the x-axis, and different spatial resolution in the y-axis. They are like electrophysiology, which is a classical one, optical imaging, magnetic resonance spectroscopy, magnetic resonance imaging, positron emission tomography, autoradiography. Well, each of these techniques have their own advantages, as well as there are certain limitations, going from invasiveness to non-invasiveness, going from different temporal to spatial resolution. To give an example, electrophysiology is one of the oldest technique which has been developed like 3 or 4 decades ago, where actually we insert a high impedance electrode into the localized brain region. We can actually measure how the neurons are firing action potential. They're called either the single units or multi-units, or the sum of activities, local field potential. Then we can interpret, how locally the brain is emitting this electrical signal during particular sensory task. But the limitation is these electrodes can pick up like few neurons, like 4 to 10 neurons, depending on either the single electrode or when you use 2 electrodes, maybe 10 to 12 neurons. And again, there is a limitation because we cannot poke the whole brain with multitude electrodes because we are going to damage the brain. And more later, the optical imaging tool was developed. This has a fantastic spatial and temporal resolution, because whenever there is a localized increase in the brain activity, there is an increase in blood flow, which causes change in oxy-deoxy hemoglobin. Both have different absorption properties, so we can actually measure the changes in oxy-deoxy ratio intrinsically without giving any contrast. But later there are some optical imaging, voltage sensitive dyes were developed where we can measure array of ions like sodium, potassium, calcium. We can measure the mitochondrial membrane potential, cellular membrane potential, influx of ions, pH, so on. But recently, from past 5 years, with the development of genetic engineering tools and molecular biology, we can actually express individual neurons or astrocytes with different colors. For example, we can express genetically modified green fluorescent protein in neurons and red fluorescent protein in astrocytes. We can actually multiplex at different wavelength, and we measure simultaneously neuron and glial activity. So like a rainbow which gives a different pattern, you can see a rainbow with different flavors of colors and simultaneously measuring the whole brain activity. It's an awesome technique. I really liked it, but the problem is the light does not penetrate deeper into the cortical tissues. It has a limitation up to 600, maximum 800 micrometer if you use 2-photon microscope. Well, the positron emission tomography is another awesome technique which actually measures the oxygen and glucose consumption, blood flow, blood volume. But one of the limitation is we cannot apply this to measure repeatedly on the same subject because it involves injection of radionucleotides. So among all, MRI stands in an ideal position where we'll have a decent temporal and spatial resolution, and we can measure by using intrinsic contrast, because the body has water has 2 hydrogen and oxygen. Hydrogen has proton is positively charged, but it does not sit quietly like that. It spins around its axis. As we know from high school physics, whenever there is a magnetic moment, they act as a tiny bar magnet, and randomly these are oriented in different direction. As soon as the subject goes into the magnetic field, actually they start persisting around the field direction. Then we just give a trick. We apply some radio frequency pulse of 90 degree and we make the spins to orient in 90 degree angle. Then we just turn off this radio frequency when they are relaxing back to the normal precision. So we actually take that information and image the brain, which is completely non-invasive. Apart from MRI, just not only imaging image, but use magnetic resonance spectroscopy, which actually measures a lot of oxygen consumption, glucose consumption, neurotransmitter flux. Recently, we are doing it for pH temperature measurements. So most of my talk today is mainly based on MRI, more specifically on functional magnetic resonance imaging. I'm just going to give you a brief overview. What does bold functional MRI measures? Here is a flow chart For example, when during a localized brain activity, there is an increase in oxygen consumption, followed by a larger increase in cerebral blood flow, which causes a change in oxyhemoglobin and deoxyhemoglobin concentration. We all know that oxyhemoglobin is a diamagnetic, deoxyhemoglobin is a paramagnetic. So they affect the magnetic susceptibility, which it affects the physical properties like T2 star signal. Then there is an increase in MRI signal intensity. Basically, when there is a neurons are firing during localized activity, larger blood flow, then oxy-deoxy changes, MRI signal goes up. This was actually demonstrated way long back by Linus Pauling. If you take the red blood cells, they have two oxy and deoxy component, and oxy being diamagnetic, they are not perturbing the magnetic flux, and whereas deoxy is perturbing. Well, over the years at Yale, I've been working on different rodent sensory models. I've established, when I came to Yale for a postdoc, this was the only model available, the FOPA, but later I developed all these three models. Now we can able to use this high field MRI to map the brain activity at super high resolution and 2 temporal resolution. I'm going to talk about these individual models in very short amount of time, but just briefly, like for example, we can connect a copper electrode to the FOPA, and we can actually image in vivo the somatosensory forelimb activation at this resolution. Similarly, rats have this facial whiskers called, the facial hairs called whiskers. We can perturb single whisker or row of whisker or the whole whisker pad. We can measure the barrel type of activation as you see at this kind of resolution. Actually, you can go much higher, but this is an example. And again, we can give visual stimulation to the rat and see the different regions like visual cortex, supracolliculus, and also different part of the brain activation during visual stimulation. And this is one of my favorite, like how we perceive sense of smell, because there is a tiny region called olfactory bulb situated in anterior to the brain. They actually code the wording, which is one of the most complicated coding, which I'm going to explain later. So we can actually map that activity glomerularly at 200 by 200. In fact, I have pushed the limit to 100 by 100, 125 micrometer recently. Very few people in the world does this thing. Yale is one of the first group to prove this, and currently like only 2-3 people in the world are doing this imaging. It is very hard to do because the olfactory bulb is only 3 millimeter cube. Shimming this at high field is really challenging. Well, just to give overview, what are the techniques I used? This is an horizontal Bruker scanner, which is 11.7 Tesla. Just to give you an overview, like what are the different field strengths currently used for clinical? Currently, like 1.5 and 3 Tesla are the main ones throughout the world people are using for human imaging. This Tesla means it's a unit of magnetic field strength. The higher the field, you get better resolution, both in terms of imaging quality and also in terms of spectroscopy. So at Yale, we have 3 animal scanners, 4.7 Tesla, 9.4 Tesla, and 11.7 Tesla. And for humans, we have a bunch of scanners. We have 3 Tesla, 2 4 Tesla, and recently we have 1 human 7 Tesla. For last 2 years, we were the highest field in the world, but now a lot of other people also got it. For example, University of Minnesota recently got approved to get human 10.5 Tesla. And for animal, the France is now like implanting a 21 Tesla animal scanner. So all of these studies were done with this high field magnet. Just to give overview, this is the bore where actually we insert the coil with the animal. And it has the drum-like structure. What you see is 2 concentric cylinders where we put liquid helium and nitrogen to keep the magnet as in superconducting state. And these are the technical details. And for the most functional imaging, I use this gradient echoplanar imaging. So this is actually revolutionized the imaging. For example, in 1 second, we can scan the whole brain with decent image quality. And just after my functional experiment, I also do some electrophysiological recording, take the animal, put it in the neurophysiology rig, and we know the area of activation. So we insert this electrode into that area, what fMRI shown, the activation map, and we measure the electrical activity. So these electrical activity were like, one is low pass, we get local field potential, band pass, we get multi-unit activity. But as I said, this is a limitation. Single electrode can pick up very few neurons, 4 or 5 neurons. For example, if you just vibrate your thumb or forefinger, you like 50,000 neurons may be firing, but it's very difficult to get the ensemble activity of neurons. In order to do that, recently, like last 2 years, I've been using this multi-array recording. So as you see here, it's a 7x7 electrode by 3 by 3 millimeter. Each electrode length is like 1 millimeter, and the tip is less than a micrometer. So it can measure along this axis. So you can implant on the rodent brain. The rats are anesthetized. We just open the skin, and then the dura is not open. See, the dura is clearly intact. We actually insert these electrodes on the dura with pneumatic inserters. We just give one shot. It goes like 1 millimeter pop, and it's there. And now with this, actually, we can record like with the 49 electrodes, ensemble of neurons, and then correlate better neuronal activity. So this is a sagittal view of a rat brain showing different tactile and non-tactile sensory inputs. Almost all of the sensory, except olfactory, all of the information goes through either this thalamus or superior coelicus to reach higher cortical centers. They cannot just directly go to the cortical center. For example, during faux pas or whisker, information first go to the thalamus and then go to the respective somatosensory forelimb or somatosensory barrel area. In case of visual stimulation, it goes to the superior coelicus and thalamus and go to the V1. And olfactory is most exclusive, and the odorant molecule from the air first bind onto the olfactory epithelium in the nasal, and then they make a first synapse in the glomeruli. The second synapse directly coming to the piriform cortex here. And recently I've shown also how thalamus is responsible. It's not a direct connection, it's an indirect path, but other senses are direct, but it acts as a relay circuit for other senses. Like traditionally in fMRI, we just give one stimulus and map the activity. But in reality, the brain is actually processing all the multi-signal. For example, you're driving a car, you are looking on the road, and you're listening to the music, you're talking, like 3-4 senses are simultaneously active. So if you're using this multi-processing, so there is a cortical interaction, corticothalamic interaction, corticocollicular interaction. So this is possible to measure with the advent of high-speed MRI. So we can apply this high-speed MRI in 1 second, we can map the whole brain. So that is what I am going to show in few examples in few minutes. This is a very first example. This is very easy to do. For example, you look at the rat, this anesthetized rat in the bore. So we put a surface coil on the top of the head, and we can image anterior to posterior like this. So in this example, we connect 2 copper needles subcutaneously to the forepaw and then give this stimulus current 2 milliampere amplitude and 3 Hertz frequency. Just to show it here, when you stimulate left, when you cut the image like this is called a coronal view. So we get activation in the right somatosensory area. Each trial were highly reproducible. When you give right stimulus, you get left activation. Also, it is highly reproducible across rats. So this is very, we can elegantly map the brain activity during a sensory task. But also, in addition, I told since we cover the whole brain, we can see which part of the brain is activated during that task. So the figure A shows the somatosensory region blue and different part of the thalamus in the atlas. So if you take the average, like 8 to 10 rats registered under the same template, we can clearly see the localized activity in the cortex and also in thalamus. You can also see how the time course of bold signal is evolving. Like, the y-axis shows the bold in percent, and this is the time. We give a baseline of 30-second stimulus, and then the bold signal increase, and then it come back to normal. Well, as I said, these animal imaging experiments are very valuable, but they have to be done under anesthesia because we are doing a lot of sound with the magnetic pulsing, and also to keep the animal immobile, you have to use anesthesia. We all know that anesthesia suppresses the baseline brain activity, but also in addition to suppressing the baseline activity, it also affects the sensory processing. So I have done like dozen of anesthetics to understand this mechanism, but here for time being, I'm only showing 3 anesthesia, isoflurane, domitor, and alpha chlorelose. If you look at here for different frequencies, we got a different magnitude of bolt responses, but each one have a different tuning curve. For example, under alpha chlorelose, you get a maximum response at 1.5 Hertz. For isoflurane and domitor, you get at 9 and 12 Hertz. In case of thalamus, it's a differential type of effect. I wanted to look more closely what is happening in neural activity. So in the same animal, when I inserted electrode in the same cortex, S1FL and thalamus, I found similar tuning curve for the cortex, the neuronal firing, whereas thalamus shows kind of an completely independent, like you keep on increasing frequency, it is always responding. This is quite amazing and interesting. In evolution, first actually the subcortical structure evolved, later the cortex developed. That's why we call it as a neocortex. So to me, it looks like there is a low-pass filter exists between thalamus and cortex, which is modulated by the state of the brain, whether awake or sleeping on anesthesia. So anesthesia is like a black box. When you go to the clinic, we know how molecular mechanism of anesthesia it works. Then suddenly at system level, it's completely black box. You lose your sense, you lose consciousness. These experiments actually give some insights how we can understand this sensory processing. Well, I want to give another example. This is the great model to study the brain plasticity. This is a whisker stimulation experiment. The brain is like plastic, right? We train the brain, use it or lose it. The more you train it, the more you use it, the more usage you get it. Earlier, many experiments were done to understand the plasticity using this model. I think depending on the different background of the people, I'll just give a brief introduction. Rats have these hairs called facial hairs. They are called whiskers. They're like 32 to 36 hairs present on each snout. They actually use it for navigation. The rats are visually impaired. Like human vision is by 20/20, rats vision is 20/600. They don't see what we see. So in evolution, they actually developed by whisking a different frequency that touch the object. For example, when they're going in the woods, actually, when they whisk at high frequency to see whether the object is like, rough or smooth. So the information from this trigeminal nerve goes into the contralateral of the cortex called whisker barrel cortex. They call it barrel because literally in those cortex, the cells look like a barrel column. Each barrel is like 400 micrometer thickness, spanning 4,000 neurons. So we can actually cut all the whiskers, stimulate single whiskers, and look at the barrel column activity by using fMRI. In this particular example, I am using 22 right whiskers stimulated anterior to posterior just by giving an air puff. Look at the activity for different frequency. You can see the barrel activity, and below is the time course how the bolt signal is evolved for each frequency. That way you can actually scan the whole brain, measure the whisker barrel activation, and similarly, we can actually see which part of the thalamus is getting activated. Just like the previous example, morely VPL was activated. Here we see more of ventral posterior medial part of the thalamus and PO were more activated. This clearly shows for different senses uses the projection from different area, going first to the ventral posterior medial, then to the cortex. In case of 4-point, go to the VPL and then go to the S1FL. So these are very valuable experiments to study the cortical plasticity. And the third example is the visual stimulation. As I said, the rats don't see what we see. But when we started this project, I actually wasted a lot of time because I was using different color of light. So I was not getting enough responses. Just to give, this is a good picture to show, like, see, this is our RF coil sitting on the top of the head here. And the rodent, this is a Longivan rat with anesthetized sitting here. To keep the eyes open, I put some Vaseline jelly and open it. This is under light off position and light off position. So this we built, actually one of our high school students built this one, this acrylic cylinder. So we can actually stimulate at different colors because we have different colors, LED and different frequency, and we can observe the bolt activation. In this example, so the rats are actually tuned to the lower wavelength. Actually, they do very good sensing of UV light, and they are sensing for ultrasound. So at 1 Hz green light, we get the visual cortical activation, V1 and V2, and also superior colicus activation was clearly observed. Similarly, we scan the whole brain, and we can see they're very reproducible. We go from trial to trial or rat to rat, for the same intensity, like 50 millisecond pulses of green light at 1 Hz, we can get these reproducible maps. And in addition, I also told that it's a different part of thalamus during visual. There's a dorsal lateral geniculate nucleus is responsible for visual information. We can see superior colicus, DLG, and V1, V2, and we can also compare the magnitude of bolt signal. Well, the fourth example is very interesting to me because I'm more fascinated by how the olfactory coding does and how we can observe our sense of smell. As I told, we have an anterior to the brain. These are called olfactory bulb. In rat, they're only 3 millimeter cube by volume, very small and tiny. Imaging this at high resolution is really pain, and high amount of engineering is required, to like shim those region and extract information from this region. But I was fascinated by the real, architecture of the sensory coding. For when the odorant molecules bind to the nasal epithelium, and they first make synapse at this thing. Look at this is an in vivo imaging showing different layers. The top is the olfactory nerve layer, second is the glomerular layer, third is the external plexiform layer. So they make their synapse only at this GL layer, glomerular layer. They activate at lateral foci and also at medial foci. This is an example when you give a banana flavor. We give 1 minute rest and 1 minute of banana flavor through our custom built olfactometer. We can get this activity pattern. So if you interrogate this bolt signal time course, whether you do left or right, you get similar magnitude of activation. And also in the medial, we have a different magnitude of activation. But what is interesting is the same experiment, if you do electrophysiologically inserting electrode in the lateral foci, look at the evoked local field potential. And in the medial, you get this evoked potential, but there is a jitter in time. So these kind of missing information can be get with the multimodal imaging. fMRI does not have that resolution to image with that small millisecond, glimpse. So this is quite amazing, like how we receive information, how we process information, how we store information, how we recollect information. If one of this information is failed, we are in trouble. So, well, in addition to like electrophysiology, I'm also recently doing calcium mapping. This recently published this paper in Neuroimaging. So for MRI, the same rat, we can use an RF coil and obtain series of slices like this. For optical calcium, we put under the microscope and we can obtain image like this. So these experiments were done concurrently. Recently, we are building a technology where in the magnet, we can use this CMOS camera to detect the changes, both fMRI and calcium concurrently. Just to give an example, these are the ethyl butyrate and methyl butyrate. One is, I think, pineapple and orange flavor. Look at trial-to-trial reproducibility in fMRI responses. Each trial, their very unique area get activated. These are both are food odors. And concurrently, like we have done this experiment 8 days after when the rats recovered, but beautifully like evoked calcium responses for the same magnitude. And nearly like 70 to 80% of overlapping occurs. This actually proves a point that even though I keep telling that fMRI is an indirect measure of neural activity, there is a nice correlation of the neuronal calcium activity measurements. Well, I was telling that in the literature, if you take any neuroscience book, most of the thalamic responses were described for other senses, but olfactory thalamus is not described very well. First time in the world, actually, we are showing that even medial dorsal thalamus actually plays an important role during olfaction. The reason is because we have a capability of imaging the whole brain from anterior to posterior. So I developed a new
Anusha?
ಹಾ ಮ್ಯಾಮ್, ಸರ್ ಹ್ಯಾಸ್ ಬಿನ್ ಸರ್ ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಂಡಿದ್ದಾರೆ.
ಪ್ರೇಮ ಮೇಡಮ್ ಕಾಲ್ ಮಾಡ್ತೀರಾ? ಪ್ರೇಮ ಮೇಡಮ್, ಬಸವರಾಜ್ ಸರ್ಗೆ ಕಾಲ್ ಮಾಡ್ತೀರಾ? ಮ್ಯೂಟ್ ಆಗಿದ್ದಾರೆ.
ಮಾಡ್ತೀನಿ. ಸರ್ ಮ್ಯೂಟ್ ಆಗಿದೆ ನೋಡಿ, ಅನ್ಮ್ಯೂಟ್. ಈಗ ಸರಿ ಹೋಯ್ತು ಸರ್. ಎಸ್ ಸರ್.
Yes.
Okay.
Can you hear me?
Yes,
Can you
sir.
hear me?
Yes, sir.
Okay, sorry.
Yes, sir.
Okay, I just lost. Sorry, I just have to come back.
Yeah.
Can you see my screen?
Yes, sir.
Okay, I'm sorry about that. So basically, we can take all of these activation maps, whether during tactile or non-tactile, and we can actually project on a 3-dimensional atlas. For example, if the red is the area for a 4-pi, you can see in the cortex and thalamus, or for visual cortex, which is the blue one. That way, you can able to map different sensory onto these different views of the brain and see which are the regions responsible for sensory processing. So, well, these are only like block design paradigm we use for fMRI, but we can also use single pulses. Like I was interested in studying how brain processes information linearly and non-linearly. So I designed some experiment going from pulses n is equal to 1 to 4, like, and also playing the interstimulus interval frequencies going from like 1.5 to 6 hertz. When I did this experiment, these are the measured bolt responses. At low frequency 1.5, going from 1 to 4, we get linear augmentation in the bolt response. Whereas when you go to the higher frequency, like 3 hertz, still you get linear, but the linearity start decreasing with higher frequency. And similar responses were observed with another contrast called cerebral blood volume. Here to the same thing. At low frequency, you get a linear increase. At high frequency, it start adapting. To view it pictorially, look at it. For a 1 pulse, just a square pulse of 0.3 millisecond, evoked a contralateral somatosensory area. 2 pulse, 3 pulse, 4 pulse, highly linear. We get this kind of linear processing or non-linear processing if you go to high frequency, the activity decreases. So I was more curious what happened to neuronally. So in the same animal, when we inserted electrode, for each pulse, the red dot indicates for single pulse, you get evoked neural activity, 2 pulse, 3 and 4 at low frequency. Whereas when we come to the higher frequency, 3 hertz, you see already the second pulse is showing an adaptation, clear adaptation. And at the high frequency, 6 hertz, only the first one gives a stimuli, the remaining get adapted. As we increase, because the repetition interval is minimized, that means the neuron doesn't get enough time to charge it and fire an action potential. That's called an adaptation. And similar thing happened with the multi-unit activity measurements, correlating very well with the LFP and bold changes. Thus, it is actually possible to actually measure the linear or non-linear nature to the stimulus versus neural activity in the top row, or the multimodal measurements of bold, CBV, and CBF to the stimulus. We go from linear to non-linear nature. Well, I will want to move on to the next second part of my talk to how we can actually measure this brain energy metabolism by calibrated fMRI. I just want to give a brief introduction without boring too much of biology here. We all know that glucose is the major energy substrate in the adult mammalian cortex. So in presence of oxygen, it gives 32, 34 ATP per glucose. In absence of oxygen, like 2 ATP per glucose. And we all know that majority of cerebral cortical neurons are glutamatergic, and cerebral cortex glucose utilization reflects the neurotransmitter action of a glutamatergic neurons. Just to show it pictorially, this is a synapse. This is a presynaptic terminal, this is a postsynaptic terminal, and this is an astrocytic process, and this is a capillary supplying the oxygen and glucose. Whenever a localized brain activity, action potential comes and the presynaptic neuron depolarizing the membrane, the calcium occurs, and then the neurotransmitter leads in the cleft. There are two things happening. The neurotransmitter in this postsynapse bind to the postsynaptic receptors, and the glial cells uptake glutamate into glutamine. This glutamate-glutamine conversion goes on. All of this require energy in the form of ATP. So where does the energy coming from? Energy is coming from this oxidation of glucose or non-oxidation. So this can be measured too, like presynaptic, synaptic, and postsynaptic can be parallelly measured, poking electrodes around the synapse. In addition to this beautiful functional imaging technique, we can also use 13C magnetic resonance spectroscopy, where actually we can label this the glucose with 13C because 12C is not excitable. So the 13C in the blood, actually the glucose enters into the Krebs cycle, as you see here in the presynaptic terminal, and the glutamate-glutamine conversion keep on going on here, and which actually generates the cerebral metabolic rate of glucose. So this V cycle and other imaging parameters can be spectroscopically measured, and we do routinely in the lab. This is slightly invasive because we have to inject some of this labeled compound and keep the animal under systemic physiological condition. Well, recently there is a renewed interest in CMR2 mapping because PET has been used mainly to measure the cerebral metabolic rate of action non-invasively. Whereas 13C and 17 were very expensive, we can also measure non-invasive. Both of these techniques have low resolution, poor temporal resolution. Because MRI has a resolution, it allows non-invasive imaging of almost the entire brain with the greatest possibilities. So the bold signal reports an indirect measure of neural activity. So it is still possible to calculate from the traditional bold signal, provided if you measure other multimodal parameters. Not to complicate things here, the bold signal S is a function of cerebral blood flow, cerebral blood volume, cerebral metabolic rate of action consumption, and hematocrit. We can actually measure bold by MRI, we measure CBF, use spin labeling. We can also measure CBV by contrast agent. Then we can actually compute the CMRO2 by using this equation. We know that A is a magnetic field dependent contrast. To have that spatial resolution to go at that micrometer for imaging, but what we did was we actually make a block here, upper, middle, and layer with different colors as shown here, which were like 600 micrometer apart. The reason is the rodent brain is like 1.2 millimeter, the whole cortical surface. This is the whole cortex, and this comes to the subcortex. So we applied this multimodal laminar responses to measure the bold, CBV, and CBF. As you see here, the time courses in the upper, middle, and lower. The upper, we get a larger magnitude of responses in the bold, CBV, and CBF. As we go down to the layers, we get a smaller magnitude of responses. This, as I said, because of the neurovasculature and type of cells projecting to each region. Later, at the end of the experiment, we inserted electrode layer by layer, measure local field potential, multi-unit activity. Then we actually computed CMRO2 based on MR data. And here is a summary showing, all of the measured hemodynamic, bold, CBV, CBF across layer, upper, middle, lower, and CMRO2 and MUA and LFP. As you see clearly, the neural activity measured electrophysiologically between like lower, upper, and middle correlated very well with the CMRO2 measure with MR ratings. So this gives kind of a very huge confidence that we can able to apply this technique and measure this energy-based information in humans. So for example, as we age, our cortical metabolism decreases and there's a cortical thinning occurring unless until you do meditation and yoga. Because it has been shown that people who actually do a regular meditation and yoga, their cortical thinning is not disturbed at all. Actually, I've seen some patients, actually some subjects, some Japanese subjects actually who are doing 40, 50 years meditation. Their brain is just like a 30 or 40-year-old had a brain. There's no thinning. But if you take like 20 to 40-year-old like drug abusive or like other kinds of problem, you can see the shrinkage of brain. So we can apply this cool technique to actually measure this layer dependent or cortical energy metabolism measurements. This is kind of a summary, just what I told. And in the, I don't know how much time I have here. So we can actually apply this. The last part of my talk, I'll show you these different animal models, neurological and psychiatric disorders. The first model is that, these models are developed based on the human disease. The Alzheimer's disease is one of the progressive neurological disorder, which is actually recognized by the loss of memory. It was invented long back, around 1901. Alois Alzheimer actually took the patient of an Alzheimer, the sections he showed that there are presence of extracellular beta amyloid plaques and intracellular microtubules. So they're called tau proteins responsible for this disease. Ever since, actually, we have not progressed much, even though so much of histology and a lot of biomarkers are available. And over the years, a lot of animal models were developed. There are genetically engineered mice models overexpressing either A beta, amyloid beta proteins, or tau, or a combination of both. And many models have been used, and some of the models work great in the experimental bedside. When we started with the treatment, it worked too, but when the treatment goes to a human, it failed. So still, it is very difficult to understand the nature of this disease. The recent hypothesis about this disease is in hypometabolism. It's like an insulin in the brain. Before distracting from the topic, I will just explain about this model. So this model is based on a rat model. This is unlike genetic. We actually use a chemical-based method. So like most of the disease is like sporadic. We don't know the cause of this disease. So this was actually developed by a Teconic. They actually use 3 cocktail of chemicals, chemically induced onset and progression of AD in a long-given rat via slow release of this sulfate, L-methionine, and also A-beta plaque based on this. This, according to them, this requires much shorter time to disease states, which is comparable to transgenic model. It takes only 4 weeks. How do they actually take this? They put all the cocktail of 3 chemicals here, and this cannula goes to the ventricle, and actually this has been designed that slow infusion occurs for over a period of 4 weeks. In this, you have to coordinate. At the end of 4 weeks, it has been shown that these rats have a behavioral abnormality. They did the water maze test and so that problem, and also histologically they found excessive amount of plaques and tau. So this is a great model. Okay, then I thought, okay, why don't we use it in MRI and fMRI and find out the structure and function, how it is affecting our sensory processing. Here is an example. If you see in the left part, this is the control rat here, and the right when the Alzheimer's disease rats. So during 4-part stimulation, like in the control, we get a very good localized activation in the right somatosensory area. In Alzheimer's rat 2, we get an activation, but the magnitude is significantly reduced compared to the control rat. If you interrogate the time course, you see the red is in the control and the blue is in Alzheimer's. So it shows there is a lesser magnitude of bolt responses in the Alzheimer's rat compared to the control rat. However, we have no differences in the thalamic region. But I always do electrophysiology in these animals, and I inserted electrode in the somatosensory forelimb and in the thalamus. We look at the red trace from the cortex and blue from the thalamus, especially VPL region. So there is an interesting thing here. See, when the stimuli onset occurs, the first information comes into thalamus at 3.4 millisecond. Later, after 5.3, you get signal in X. Actually, this is a temporal delay or the good information, how it is being sensory processing in the location. So when you make a histogram of the spike count in control versus AD, you see very clear differences here. Magnitude of neural activity is clearly decreased in Alzheimer's animal. Not only the decrease the magnitude, it is also affected the latency. My hypothesis for this, or my interpretation about this data, is like because of the places are present, they affect the neuronal communication. That's why the neuronal wiring changes and vascular coupling is disturbed here. Well, there's another way of looking the activity data. It also shows clearly the magnitude of these neuronal activity in AD rats compared to Alzheimer rat. Well, in summary, these results suggest the correlated functional deficits in the cortex for hemodynamic and neural responses in AD rats as compared to the control of the same age and strain. But again, I wanted to apply further this calibrated fMRI tools to study the energy metabolism, what happens actually locally, why there is a differential effect in the cortex or thalamus or hippocampus, those kind of things we can study using this. I wanted to discuss the second model. This is a MAM rodent model of schizophrenia. Schizophrenia is a debilitating neuropsychiatric disorder typically diagnosed from late onset in late adolescent to adulthood of around like 17, 18 years old. There are like positive and negative symptoms. The subthreshold behavioral symptoms often precede the diagnosis of schizophrenia. However, these prodromal symptoms have not been consistently associated with either structural and functional brain biomarkers, thus limiting the chance of early diagnosis of schizophrenia in clinic. So we were looking for this model anyway, like, of course, like the one of schizophrenia is hallucination. We cannot ask that whether it's hallucinating or what it is doing, but I wanted to find out some of the structural and functional biomarkers. So the way this model was developed was they take this adult pregnant rat, and they inject this chemical I'm unable to pronounce this properly, methyloxymethanol acetate, MAM, a potent genotoxin that methylates nucleic acids and alters the neuronal differentiation and migration, and may have behavioral impairment that is similar to individuals diagnosed with schizophrenia. This is quite interesting. For example, this is injecting at gestational period, I think 17. Like each day has a different impact. If you inject the same drug, a MAM, at 16 or 14 days, they develop epilepsy. And at 12, they develop some kind of different brain abnormality. That means during the pregnancy, the brain are growing rapidly, enormous amount of speed. So this kind of, the timing is very important. Actually, we use this multimodal MRI measurements to measure the anatomy, metabolism, and function, and were screened for early biomarkers in these MAM rats and compared with the same with saline-injected control rats. Actually, we published this 2 years back in Biological Psychiatry. Just to show some of the important finding here, the brain morphology changes in MAM rat compared to the sham rats. The first one, the figure A, shows the visual cortex here. You see the mark in V1, this is the cortical thickness we measured. So the red one shows the MAM, the schizophrenic rat, and the blue is the sham, the saline injected one. You see a significant decrease in the cortical thickness in the schizophrenic rat compared to the normal rat. Not only the cortical volume change, there's also changes in the ventricular volume. You see the ventricular enlargement occurred in the MAM rats compared to the sham rats. Then there's a large ventricular. There's a ventricular enlargement happening in the posterior region of the brain. Well, we can also use a different technique called structural connectivity. For example, you see the directional encoded maps here. The fibers from left to right brain are connected through axonal connection. You see the red one for horizontal, vertical is the green one, and the blue is going across. So here too, we see clear differences in the fractional anisotropy in the MAM and sham rats, which is actually validated by using the histology, where say that axonal density changes. That means increase in axonal connectivity. This is quite similar thing found in the human subjects in the clinics. So we found a structural, in this model, the structural changes were similar to the human what they found in clinic. Then finally, we wanted to see what is happening to the resting state functional connectivity. I'll just give a 30-second introduction about this. Like so far, I talked about the functional, like task evoked MRI. You give a particular task and you see evoked response. But there's another thing, you can actually use it to measure the resting state functional connectivity by using resting state fMRI. That means the subject will not do anything, goes and lies in the scanner, and series of images are obtained for like 6 to 8 minutes. Then we take the image, then put a voxel on the right side of the cortex, and we get a time series. Then we start correlating across different regions of the brain. Very interesting things were observed. For example, when you put at the right side of the cortex, the left side of the cortex correlated very well, showing that these neuronal fluctuation oscillations are like, correlated well to the well-connected regions. We all know that left and right brain are connected through this corpus callosum. That makes sense, but we still don't know the real science behind this functional connectivity. Now it's kind of used in the clinic as a biomarker, but a lot more to dig about it. So I thought, why not use this one? So in the same animal, we connected all of these resting state scans. So as you see here, we put a seed in the visual cortex and start correlating across the different regions. The x-axis here is going from V1 to U1 amygdala. You see the higher connectivity strength in the MAM rats compared to the sham rats. The kind of hyperactivities were observed. So these hyperactivities actually correlate, to my sense, to the other behavior. And also different regions, for example, the seeds were located in orbitofrontal cortex, metasensor cortex, and dorsal hippocampus, A, B, C, as shown here. We find something interesting in the orbitofrontal cortex. MAM show actually differences, a significant difference across other regions compared to somatosensory or dorsal hippocampus. This kind of showing there's some connectivity variation from orbitofrontal cortex to the other region of the brain in schizophrenic compared to the normal, right? So to summarize this topic, the MAM rats had lower or higher structural connectivity in anterior posterior corpus callosum. However, the orbitofrontal cortex of MAM rat showed a lower resting state functional MRI and functional connectivity. It's called hypofrontality. So in contrast, functional connectivity measures were all higher in the visual cortex of MAM rats, posterior hyperactivity, which might parallel the perceptual problems in schizophrenia. Well, and the last model I wanted to just show is about this mild traumatic brain injury.
ಬಸವರಾಜು ಸರ್ ಮ್ಯೂಟ್ ಆಯ್ತು ನೋಡಿ, ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿ. ಬಸವರಾಜು ಸರ್ ಮ್ಯೂಟ್ ಆಯ್ತು. ಬಸವರಾಜು ಸರ್.
Yeah, sorry. Can you hear me? Hello?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Okay. I'm sorry, this is happening. I don't know why. I'm touching some button here. Can you see my screen?
Yes, sir.
Yes, sir.
Okay. So yeah, so when we are playing, like there's a mild concussion, we actually neglect it. So when we basically after the injury, it takes like 20-30 years to develop some kind of an abnormality. People get like stroke or epilepsy. So in order to mimic that, we use this early life traumatic brain injury models to identify the morphological and functional markers underlying this developmental plasticity. So previous studies in rat model of developmental TBI show that there are deficits in persisting through adolescence, including sensory motor impairments, locally decreased baseline CBF, and globally altered above blood flow. So actually we use this nice model. It's used by lateral fluid precursion injury model. We did a surgery and then we drop a pendulum for a very short amount of time in the injury created rats. Whereas in case of a sham rats, we don't drop anything there. We just open just for the surgery and then we close the skin. So after the injury creation, we started observing the behavior of the animal. Then in 3 months time, then we do this multimodal MRI. We do task resting state fMRI and DTI to measure both structural and functional MRI. Here are the data. To see the structural connectivity, look at this one, A. It is comparing the sham rat, non-injured versus the TBI, traumatic brain injury one. So in the T2 gradient, we don't see much injury because we drop into the cortex. If you look at the spin echo image, clearly there is an injury here. This is actually measured, I think, 10 weeks after the injury. And also in DTI, you can see how the corpus callosum is and this side of the brain is damaged. So we can able to actually map by using the diffusion tensor imaging, differential fractional anisotropy and apparent diffusion, like in diffusion of water molecules are changing in the TBI compared to the sham rats. So why not study functional connectivity? So we compared the resting state connectivity in the sham rat. For example, if you put a seed in the right side of the cortex, we can see very good left to right correlation in the activity map. You see very clear correlations are there. But whereas in case of TBI, we lost the connection. Same experiments were performed in the injured one. Since there is an injury here, because the brain is connected left to right through the corpus callosum, we lost it. This clearly shows there is something happening at the network level. And we are also interested in looking at the hippocampal network, and here too we see a very affected area in the hippocampus, whereas thalamus did not show much effect. Now, then we also did what's called cerebrovascular reactivity experiment. That means like giving a global challenge to the brain. Here we give 5% carbon dioxide to the animal and measure the evoked bold responses throughout the brain. We are clearly seeing across different regions of the brain, like cortex, dorsal hippocampus, and ventral thalamus, a decrease in the magnitude of evoked blood flow responses. This is kind of indicating that in injured animal, even the neurovascular and metabolic couplings are affected. So, but as I say, these models are helpful for the drug treatment or development. So we were actually in collaboration with one of my good friends, Sridhar Kanupati. He's in New Jersey. He was using this camphorol, a mitochondrial calcium importer blocker. He actually kind of shows that if you protect mitochondria, you can protect the cellular energy budget. So this camphorol is a flavonoid. Then we use this to, like, we create an injury in the animal, and then we started treating after the injury with this drug called camphorol. And we measured all the structural, anatomy, DTA, and also the functional connectivity experiments. Here are the results. So if you see figure A, and so the red is the injured animal, and the purple is the treated one. So the fractional anisotropy increased. And actually, the ADC also significantly impacted in this animal. And more significantly, the resting state connectivity, look at this one, in the TBI plus vehicle, we don't see any differences, because here we just give no drug here. So in the TBI animal, the connectivity is lost in the A, in the second column. Whereas the animal treated with camphorol, you see the connectivity came back very significantly. This was recently published in Journal of Neurotrauma. So this gives like more confidence, like we can actually monitor in time how these drugs are responding to the treatment and what are the physiological and network level changes are happening in the brain. So in summary, this developmental TBA was sufficient to induce diffuse deficient observable in even up 2 months later, 8 to 10 weeks. So impairments were detectable at the level of structural fiber integrity, functional hemispheric networks, systemic cerebral vascular reactivity. So this model of local and diffuse developmental injury provides an ideal translational platform to the development of treatment beyond neuroprotection. So in future, actually characterizing the nature and underlying neurobiological causes of psychiatric and neurological disease at neuronal circuit and network level has proven to be less than straightforward and necessitate the again, animal image in combination with other in vivo modality techniques is necessary for understanding the mechanistic understanding of function and disease. I just give like 2 minutes introduction on this. There are new techniques available called optogenetics, DRED, and pharmacological MRI. So optogenetics is a relatively new technique where actually we can express different genetically modified animals. We can express different neurons and glial cells with different light. We can shine a light on the brain and obtain what is happening on fMRI. So simultaneously doing this is called opto-fMRI or DRED. DRED is actually stand for designer receptor exclusively activated by designer drugs. You can actually express these kind of specific in the hypothalamus, for example, hunger. You inject an agonist, animals start going and craving for food. So you can actually induce this and look at in real time scale at fMRI what is happening throughout the brain to understand the obesity or other mechanistic view of diseases. And the other one is the straightforward pharmacological MRI. You can inject any drug and see how the drug is responding globally in the brain. So basically applying this neurovascular and metabolic coupling in aging and neurodegenerative disorders, I am more interested in actually looking at olfactory dysfunction as a biomarker in aging with high resolution structural and functional imaging. See, in aging, we lose sense of smell, and it is very pronounced in Alzheimer and Parkinson. People lose almost 90% of the smell. People don't give importance for the smell loss. I think recent COVID, people are losing smell. We don't know why. That's quite exciting. So I am actually applying this very novel biomarker to see how we perceive, because the odor identification, differentiation, discrimination changes as we age. Again, like, taking all these things, we are so lucky to have such wonderful engineering, computation, advanced physics. With all these efforts, we are trying to image this thing. Imagine our ancestors like Aryabhata, Bhaskara, Charaka, Shushruta. They have done such a fantastic work, and they've always told that mental fitness is more important than physical fitness. That means ahara, the food we eat, vihara, the exercise, and vichara, the thoughts. All of these things are contributing to a better mental health and physical health. And we have to give importance for all these things and respect those ancestral discoveries and trying to move forward. Well, I did not do this work alone. I have a wonderful colleagues at Yale in collaboration with Famid, Peter, Hal, Neurology, and Justice. Gordon is one of the pioneers, around 87-year-old. He has written a lot of neuroscience textbooks. And a lot of high school students to postdoc to graduate students who contributed to this work. Unfortunately, I cannot acknowledge all of them, but thanks to their work. And to the all NIH support, and to my wonderful kids who always inspire me to do more challenging work all the time. And thank you. And if you have any questions, I'll be happy to take.
Thank you, Basavaraju, sir, for sharing your expertise and experience in the field of neuroscience. Now the session is open for discussion. Sir, actually we are receiving a lot of positive reviews, and there is a question. Should I repeat the question, sir? Basavaraju, sir?
Yeah, please ask the question, yeah.
Does deficiency in calcium causes any change in neuron work?
Well, actually, that's a great question, because if you take in a neuronal cell, the cellular calcium concentration is 100 to 120 nanomoles. The extracellular, which is very high, 1 to 1.2 moles. Intracellular calcium is very small. So this calcium is highly titrated, actually, which always keeps the cellular insides concentration at 120 to 180 nanomoles. But during like aging or other neurological disorders, there is a huge changes in the cellular membrane potential that causes a large flux of calcium. This, if the calcium is not sequestered back and forth, it actually the cell uptake a lot of calcium. This mitochondrial calcium overload occurs. Whenever the mitochondrial calcium overload occurs, actually it does not synthesize ATP properly. Apart from, there is another dangerous thing happening, what is called apoptosis. What it does is it actually releases the cytochrome C from mitochondrial to the mitochondrial transition 4, which combines with a lot of apoptotic activating factors and leads the cascade of events over time. That is one of the fundamental things for neurodegeneration, very important. So.
Hello. Thank you, sir. There's another question. If rat is anesthesia, will they identify color? Light?
Of course, yeah. Yeah, because basically what happens is anesthesia, we can observe. Basically, the anesthesia is a very complicated story. Anesthesia suppresses the brain activity. Some light we measure, like under anesthesia, we're going from 1% to 1.2% how brain activity changes. But anesthesia does not affect the sensory processing in the sense the light actually here is the photon impinging on the retina actually are transduced into electrical impulses and the signal communication occurs. So one of the thing what I was saying is this in the visible spectrum, they don't see, like they are more tuned towards the ultraviolet light. The blue and green are very good. If you go to the red, actually we have shown it when for the same intensity of light, the red color has a very poor vision compared to the blue color. And actually, I'm intending to do some UV experiments. There is one student actually working on building this UV apparatus to send the signal, but animal ICOOC committee is like kind of not approving it, but we are fighting against it, yeah.
Okay, sir. The next question is from Anita. Is the diabetes affects neuron? Septic signals.
Actually, I had
synoptic signals. Sorry.
Yeah, I have done a studies. I did not show the slide. Basically, what we do is we do a clamp, hypoglycemia. Normal glucose is like 120 to 140 milligram per deciliter, right? So when we have normally when the food intake is proper, when you measure the glucose in the blood, that is the level. So what we did an experiment is under normal glucose, we measure the fMRI. We give a stimulation, we measure the activity, just like a control. We got a good activation map. Then if you clamp with insulin, we can actually titrate the glucose level. When the glucose level goes to 60 milligram, then the activity is still there. But when it goes to 40, then it lost the evoked fMRI bold signal. But when it goes below 20, then the animal started seizing with epileptic seizures. So then we did the reverse engineering. When we started giving back the glucose, then the animal came back to normal. So we can actually do these kind of experiments to identify what's happening when glucose changes in vivo.
Okay, sir. The next question is from Veena Etish. Like color detection in brain, is it possible to take the image of taste and flavor of the food detection by human brain?
Yeah, of course. Actually, you have to read my recent paper I published this year about orthonasal versus retronasal. Actually, we perceive sense of smell through two routes. One through nasally, which is called orthonasal. Other, when we take the food molecules, when we are chewing the food, the odorant molecules dissolve and go and affect the olfactory bulb, which is called retronasal, which is actually good perception because we combine taste and smell. For example, when you drink a wine, actually you're not smelling it. The thing is going from your tongue, the taste to a different mechanism and activating olfactory bulb. There's a new book called Neuroimaging, which actually describes how I actually perceive the flavor perception. So the brain is actually like we are experiencing and we enjoying multisensory, both taste and smell. For example, during your cold, if you lose taste or smell, right, you don't enjoy one of it. Both has to be there to enjoy the real flavor. So actually, I've shown it in an animal model during retronasal smell versus orthonasal smell, how this taste and smell, odor and mapping changes in the brain. Yeah.
Okay, sir. Madam Gayatri MP is asking about brain imaging in hyperactive kids. Are there any markers in
actually a lot of people are doing this? Yeah, ADHD, actually attention deficient hyperactivity disorder. I don't know who coined the term. There is nothing like that actually, because this guy who coined this term, because see, when we were kids, we were jumping from tree. Then they look at us, we are all ADHDs, right? So this is somehow it actually, kids are active all the time. Somehow there is no experiment has shown these kids have higher brain connectivity or different region of the brain have some kind of an activity. But I am a biggest opponent of English medicine treating all of these things. If any of the psychiatry people are watching this, they hate me. But that's the kind of issue now. In US, it is very common. This ADHD has been diagnosed kind of, if the kid is like hyperactive, a little bit active, they call it hyperactive, and they diagnose and they'll have some concessions. Yeah.
Okay, sir. The next question is about neuron transfer, sir. Neuron transfer person is possible. Person to person is possible.
No, I think there are a lot of stem cell. I think the last speaker of this one is very expert on talking. He's working on a stem cell. He can give a good light on this topic. He's working on InStem. So the stem cell therapy is being like working now, so the neurons don't regenerate, except in like olfactory bulb, they have a regeneration. But in different part of the brain, it's difficult. So they are using this InStem, the stempeutics, I think they call, or stem cell therapy they are using. Yeah.
Next question is about rats. Rats uses their all legs for different work, but human may be right-handed and left-handed. Is this affects the activity and thinking of human brain?
No, basically, like I was showing an example about the mild traumatic brain injury, right? So whenever we cause an injury into the right side of the brain, the left forepaw is like, it's not started using, the left forepaw region is affected a lot. But over the time, he started using, but it will not come to the 100% reuse. As I said, the brain is like a plastic changes, right? The way you use it. Some people get adapted to using right hand. From the beginning, you start using your right hand, your left cortex will be more active. If you use right, left is active. Actually, that's actually a very good question, because recently we were showing for Alzheimer therapy, Alzheimer people is like losing memory, right? So the brain is like use it or lose it. The more you use it, the more the neurons are active. So we always traditionally brush with our right hands, or left temporal lobe is active, and then we are telling that use your left hand also so that right is active. You should start using left and right both to keep the thing more active. So rodents have been evolved. Actually, they use, we can do a very good experiments. Like, for example, you damage one side of the brain region and how it start adapting to the other limb, and how those cortical regions are wiring is changing, electricity is changing, those kind of experiments, yeah.
Okay, sir. How is vertigo different from seizures and epilepsy?
What is different? Sorry.
Vertigo different from seizures and epilepsy.
I'm not expertise on that. So I'm sorry.
So we can take the another last question, sir.
Yeah, sure.
So when person have high fever, he will suffer epilepsy. It may affect brain. So the person become unconscious. Which part of the brain affects? What steps to be taken immediately?
Oh, I'm not a medical doctor to answer that. It's a very clinical oriented question. But I can tell, like, we have done a lot of epilepsy experiments. I have some data to do that. What we do is we simultaneously measure the EEG and fMRI. When the animal brain is seizing, we can see the EEG going on. During that time, we can actually measure the functional MRI bold activity. There are some very good interesting things. Some part of the brain is having higher seizure, where you can see a very good positive increases in the fMRI activity. Whereas in some part of the brain, there's a negative, there's a decrease in the bold activity. This is very interesting because there is not only like there is a vasodilation, whenever the neurons are firing, the blood flow rushes in, there is a dilation, where you get a positive bold. In some cases, some subcortical regions, vasoconstriction, the blood vessel shrinks and the bold value goes down. So a lot of experiments now we are doing, implanting electrode into those regions and seeing during epileptic discharge that we cannot do it in humans, right? We cannot poke electrode into all over the brain, but in animal we are implanting these electrons, understanding the neuronal dynamics during seizure state. But those the clinical question I should not, even though I know a little bit, I should not answer because I am not in the medical doctor. So.
Hello.
Yep.
The last question, sir. Shall I?
Yeah, sure. Yeah, no problem. Yeah.
Yes, ma'am. Some participant.
Hello.
Okay, vitamin B12 have any impact on neuron growth?
Actually, there are so many things. Actually, if you look at literature, there are people are dumping so many things now to study the effect. Like there are free radical therapies, like vitamin therapy, all of these things. But there is no clarity actually how it does. It depends on this system requirement, right? Sometimes I am actually again biggest opponent. Here in America, that is very common. Like when you go to, there's a vitamin shop, everything there is vitamin. Then an adult American take like 20 tablets, for everything, for all kinds of things. But to me, I feel natural thing. Eat vegetable food. Eat a lot of like fruits, vegetables, greens. These are the things. See, for example, like, the lemon is such an antioxidant properties. You drink, instead of drinking 10 cups of coffee, 1 lemon water a day keeps you so healthy. So it is highest in antioxidant, vitamin C, or actually the best is the white guava, that has the highest level of vitamin C or ascorbic acid. Instead of taking this 100 milligram of vitamin C a tablet, eat 1 guava. That's natural, good fiber and good vitamin too. Yeah.
Some participant wanted to ask a question. Any participant?
Hello, ma'am. Can you hear me?
Yes, Jyoti madam.
Sir, I have a question for you, sir.
Sure.
Sir, it's regarding ethical issues, which is not touched upon. Neuroimaging studies involving human subjects can raise a range of ethical issues, sir, related to informed consent or risk proportionality, confidentiality. Can you please tell something about that?
Yeah, sure. Any studies, whether animals or humans, you have to go through a review board. Even actually doing animal experiment is more tougher than human experiment because we have this IQ committee. Like every simple protocol has to be like approved by this committee. It's a very strict and they are very vigilant. If you actually, they come and check your experiment. For example, when you are experimenting, they come and check like 3 months old record. Like when the rats are lying in the scanner, we have to, just like a human patient, we are measuring the blood pressure, we are measuring the temperature, we are measuring the systemic thing, like everything is going on. Under anesthesia, you should not take it for guaranteed that you give anesthesia and animal is happy, and you have to just monitor just like a human. So similarly in human, there are set of questionnaires. Actually, IRB gives questionnaire that you have to answer all of them. You have to pass it. Then only Excuse me. Hello.
Yes.
Yeah, there was some disturbance. Somebody asked something else. Yeah.
Thank you, sir. Thank you.
No problem. Actually, if anybody has question, actually you just Google my name and there is my email available, and you can actually throw me a question and be happy to answer. If anyone has some important thing to discuss.
Okay, sir. Thank you, sir. Thank you for your time, effort, and lot of knowledge sharing. And thank you, participants, for your active participation in the discussion.
Hello.
Somebody is saying something.
Yeah. Can you hear me? I'm Nandish here.
Hello, sir. Yeah.
Yeah, I'm the. Yeah. Good evening. I think good morning for you.
Yeah, that's fine, sir. Good evening for you. Yeah.
Yeah. I'm the honorary secretary for this group of institution, SJR group of institution.
Nice to meet you, sir.
Yeah. Thank you. Nice meeting you. You have come from Yale University.
Yes, sir. I'm sitting in Yale University right now.
Yeah. Nice to hear, because my brother-in-law studied in Yale University in 1960s.
Oh, okay.
He was one of the architects who studied in Yale.
Oh, okay.
To India, and he was one of the chief pioneers of construction of this Vidhana Sauda and St. John's hospitals and Kandivas Stadium. A lot of buildings he designed after coming from US. So
actually, last 3 US presidents are from this university. Barack Obama, Bush, and other. Yeah.
Yeah, very good. My cousins are staying in Chicago, Purdue, and all. Yeah, Purdue. And nice to meeting you. Yeah. It's a good topic. What you spoke about the brain. Neuroscience is very good science. As you lastly said, what we require is not the medicine. What we require is the body.
Right.
Because brain is the CEO of the body.
Right.
And it always sends the signals to all over the body. Right.
No, actually, people only concentrate about physical fitness. My suggestion is to do mental fitness, right? If you keep your mind right, whole body will be all right. So that's the thing. So you have to discipline your brain. Actually, we have to learn from our ancestors, right? They have a very good discipline of life, right? Even take this COVID pandemic, right? When you come into the house, you wash your hand, and we are so much samskaras. Nowadays, people like come in boot and then go all over the thing. So we are going back to the history. So I'm always mesmerized with the kind of tool, without any kind of tools, how our ancestors discovered a lot of scientific things. Actually, I was talking to Prema Madam about Anubhava Mantapa. If you look at it, right, so much of science has been done, like Charaka, for example, invented so many diseases. He never named one disease as Charaka, but you take Parkinson, we call Parkinson Alzheimer, we make one diseases. In heritage, we have to read it properly. We are just boasting with supercomputing and advanced computational knowledge. Oh, we know so much, but actually we don't. We are just going back to history and start learning how they actually understood all these things at that level. Yeah.
The samskara is important.
Right. Yeah.
That's right. Yeah. Now, as you all know, India has become a diabetes capital of the world.
Right.
And US is becoming the cancer capital because of the malnutrition going on.
Right.
As you rightly said, what we are having, the food about 100 years back is not available right now.
Right.
Everything is depleted soils.
Actually, now they are proving that intermittent fasting is one of the good therapy. For example, they were telling Vanda Tundavanu Yogi, Erada Tundavanu Bogi, Mura Tundavanu Rogi, right? We keep abusing our food. We eat breakfast, lunch, evening snacks. How much time, how much of free radicals are generated? You look old, actually. So instead, get up at 5 o'clock, run 5 miles, do some yoga. You have to keep our life in a controlled, disciplined way. Actually, India, I don't know why, the obesity, cardiac and diabetic problems are much higher in India. And secondly, the doctors are not treating kind of empathetically. The thing is that you go to a clinic and they jump you. All these numbers, 120 or 130 milligram is coming based on the standard statistics. 180 may be good for you. You can adopt. For example, I grew up in Siddhaganga Math with Swamiji. So I saw his lifestyle. I was with him for 8 years. I saw he was so dynamic. At the age of 102, he was walking 40-year-old guy. So because he was eating only 2 meals a day, that's it. And he had always a clean thoughts, and these people live longer.
Yes.
That's one of the.
Yeah, that's right. It's true.
Yeah.
only 3% successful people in the world who gets up early in the morning. Only 3%.
Exactly. Yeah.
97% are all not there because their life is different. So you have
No,
to.
actually, as I said, no, this, like I still don't believe this 8 hours prescription of Western way of sleeping. You can yoga nidra. You can only sleep 3 hours. You can still be active, but you know how to do that. Actually, it's a discipline because the brain is active all the time, whether dreaming. It doesn't mean that you sleep for 8 hours, watch your cell phone or do thing. That is not the sleep. You sleep means you go, you die, right? When you get up, you're up. That's it. Only 2 states. One is, you know. So we need to practice that yoga. You know that's. I'm a firm believer of yoga because that's how you know, mentally you are fit.
Yeah. That's right. Yeah, actually, I practice 1 meal a day. Actually, almost afternoons.
Yeah, actually, intermittent fasting is very good
in my body. So I know how much my body can take it as per my BMR.
Correctly. The new area of research is gut-brain axis. Recently, we are actually see the gut as a second brain, right? So there are several million neurons in the gut. Actually, when you eat a banana, you feel happy because banana has serotonin, right? Even heart has neurons. You know, heart has 50,000 neurons actually. So like you keep your gut clean. For example, you eat a banana, it goes out in like 3 hours. It digests fast. Go eat some other garbage like meat or something where Western people eat. It takes 72 to 100 hours to go out of the system. And that's a problem. Anything it doesn't go out of your system, it creates a problem. And also again, the way it is cooked and the way that is consumed, you know, that I'm not going to be like talking religiously, but that is how our body responds it. What you eat, what you are. And
it's true. The blood pressure and the sugar thing comes from magnesium and calcium because of the sugar deficiency of mineral, magnesium and calcium, and these minerals are deficient to the body. That's why BP levels goes up.
Correct.
Yeah.
That's what they say. Like, you know, as soon as you don't drink coffee because pitta will increase according to it. That's true, right? Caffeine is actually kind of affecting. Instead, drink like clean water. You know, that's the way you start with. Yeah.
Most of the obesity is killing
everywhere. Throughout the world. Actually, you should see US. Like people are living longer because we are medically well controlled, right? So here people take a very good health care because all health insurance will take care of it. But somehow, like I don't want to live like that. At the age of 70, I don't want to have Alzheimer, right? Your brain is not working, body is good. So what is the use? You had to use your brain. You should be like, even at the age of 70, you should be like 30. You should feel like 35. I don't know. I am feeling like I'm young. Actually, I'm feeling like I'm studying in SJRC like 15 years ago. I still have the same thought. I always feel that I'm a good student. I always learn. So, you know, that's the way you should feel. Not that, oh, I'm old. Nothing. Actually, people are starting their career at 65. For example, like Trump is starting his career at 72. Obama retired at 55. It doesn't matter, right? So we have to feel ourselves always in a positive thoughts, and that's I feel up from what science we do. Yeah.
Sure. It's a pleasure meeting you.
Nice here, sir. Thank you
for giving your thoughts and information for Iran.
Yeah, happy to share because I'm the proud alumni of SJR College because I was so lucky to have wonderful teachers. I still remember all of them.
And I'm also alumni of SJR.
I had all the great teachers in physics, mathematics, and electronics. I still remember, like recently I was doing some electronics in the electronics room I was building. That Dr. Loknath has taught me something. It just popped up in my mind. Like I'd learned like 17, 18 years ago. They're wonderful teachers actually. So they inspired actually to do a lot of research that time. As Dr. Prema Madam was saying that people are like having inhibition to go to the basic science, right? So I was a postgraduate in physics. I never dreamt that I'd be doing biology one day. You know, it's an accident, right? So now there is nothing like this area because we are using all multidisciplinary physics, chemistry, engineering, advanced computation. Everything if you want to learn, go learn chemistry. Right now, look, I am doing mostly like all biology based, but I'm using all the learned physics, engineering, all of these, basic fundamental things. Yeah.
Very, very nice. Because I'm also alumni, now I'm serving this institution as Honorary Secretary. I did my commerce there and completed my law also.
All right, thank you. Yeah.
Thank you, Prema Ma'am. You can take over. Manjula Ma'am.
Thank you.
Thank you, Nandi sir.
Thank you.
Thank you once again, Dr. Basavaraju sir. Now I request Srimati Kalpana T.S., Principal, SJR College for Women, to give the presidential address. Over to Kalpana, madam.
Okay, I have unmuted.
Over to you, madam.
Yeah, thank you, Manjula. Ellarigu namaskaraಗಳು. Initially, I would like to wish all the success for the 5 days faculty development program. And respected speaker of the day, Dr. Basavaraju G. Sanganahalli. We are really proud of you for 2 reasons, sir. One is you're the alumni of our college, and we are also proud to see a Kannadiga Indian being neuroscientist at American University. And sir, as we know, as biology teachers, we know that brain is the most complicated organ with complicated structures and complicated functions. Delivering all these complications, you have chosen to be the neuroscientist, and really lot of appreciations to you for that, sir. And we have with us our Secretary, sir, Honorable Secretary, sir, Shri N.R. Nandish. Sir is always encouraging, cooperative, and always is highly supportive in all our endeavors. He is a techno-friendly person, and I welcome you from my side, sir. And we also have with us, I can say, most vibrant and enthusiastic principal of SJRCW, Dr. Prema Siddharaju. And she has organized series of webinars during this lockdown period, and congratulations to you, madam. And we also have exuberant participants of the August gathering. The session was highly interactive and informative, I can say. And on the outset, I sincerely thank our management and principal of SJRCW for providing me an opportunity to preside over the function. I consider this as an honor to me, madam. I would like to appreciate Dr. Prema Siddharaju for organizing Multidisciplinary 5-day International Faculty Development Program to enrich knowledge of faculty in various disciplines. Today is the first day of the program, and Dr. Basavaraju sir has very effectively conveyed information on brain imaging. So as we know that brain imaging is done by the data acquired by certain techniques, and sir stressed more on functional magnetic resonance imaging. And we know that there are 2 types of brain imaging: structural imaging, which is done to visualize anatomical properties of the brain, and functional imaging, wherein the brain areas are identified based on the task performance. So functional imaging techniques are based on physiological aspects, maybe it's like metabolism, blood flow, chemical composition, etc. The imaging of brain can help to detect brain disorders associated with problematic children, maybe children suffering from epilepsy, mental disorders due to cortical malformities. So this may help to adopt differential teaching and bring these mentally disabled children to mainstream. Sir's area of interest is different rodent sensory models of cortical and subcortical activation in rat brain by functional MRI. He has put a light on the fact that imaging helps to identify drugs for brain malfunctions, and he has briefly mentioned about activity in the synaptic region. Dr. Basavaraju sir is more concerned with functional magnetic resonance imaging to map brain activity. Sir has given technical details and how anesthesia works at molecular level. The way Dr. Basavaraju showed proficiency in speech shows that he is deeply involved in the research activity. He has also explained about the visualization in rats, olfactory sensation in rats, and other aspects of his experimental work. I thank Dr. Basavaraju for delivering such an elaborative and informative talk on functional brain imaging. I also thank our management and our Honorable Secretary Nandi Sir for his enthusiasm and active participation in most of our webinars. I also thank Principal of SJRCW, Dr. Prema Siddharaju, for providing such an opportunity to all of us. I would like to resign with a note of thanks and thank you everybody. Thank you.
Thank you, Kalpana Madam. Thank you, Kalpana Madam, for adding value to this session.
Thank you.
Now we are at the last segment of the session. I request, am I audible? Yeah. So I request Srimati Shilavati, Assistant Professor, Department of Mathematics, to propose the vote of thanks. Over to Sheila. Sheila Madam.
Thank you, Ma'am. It is my privilege and honor to propose vote of thanks in this multidisciplinary 5 days international faculty development program organized by Science Forum Scientia of SJR College for Women. Our sincere gratitude to today's resource person, Dr. Basavaraju G. Sanganahalli Sir, neuroscientist, radiology and biomedical imaging, Yale University, New Haven, Connecticut, USA, for his very effective, informative, and excellent presentation. Thank you, sir. I would like to thank Shivprakash Sir, President, N.R. Nandi Sir, Secretary, and C. Natras Sir, Treasurer of SJR Education Society, for their constant support. Thank you, sir. I would like to thank Professor T.S. Kalpana Madam, Principal, SJR PU College for Women, for her presidential address. Thank you, Madam. My sincere thanks to Dr. Prema Siddharaju Ma'am, Principal, SJR College for Women, for her support and encouragement in organizing this 5 days faculty development program, and also for her wonderful keynote address. Thank you, Madam. I would like to thank all the faculties of SJR College for Women. Last but not the least, I take this opportunity to thank all our participants from different colleges, universities, for attending this webinar, FDP, and we hope all of you enjoyed this session. Thank you for joining us. Thank you, one and all. Have a night. Good night. Thank you.
Thank you, Sheila. I request all the participants to unmute their video for a few seconds for the photo session. Please,
yeah,
please, madam.
Yes, ma'am.
Are you done with it, Anusha?
Just a second, ma'am. 1 second. Kindly hold it.
So before we end the session, let me share the guidelines. The feedback link is kindly fill and submit the form. And there is no day-wise issue of participation certificates. It will be issued all the sessions. And tomorrow, the session will start at 11 a.m. Tomorrow, the session starts at 11 a.m. Kindly cooperate. Thank you all once again. So now I request the permission of the resource person, Srimati Kalpana Madam, Dr. Prema Siddharaju, to end the session. Shall we end the session, Madam? Huh? Shall we end the session, sir? Thank you very much, sir.
Okay, bye. Thank you. I really enjoyed it. Yeah, bye.
Thank you, sir.
Thank you, Dr. Basavaraju.
Thank you, madam.
Thank you, Kalpana. Thank you, Prema madam.